Okay, I'm sitting in Harold Hillman's palatial establishment. It's the 11th of March, 1996. And I've got a whole list of questions here. And I think I'll just go through them in the sequence. This was not the sequence we arranged before, really, but it's because there's such a large amount of work to cover. What I want to do is the following things. I'm going to start rather orderly by listing what I consider to be weaknesses in his presentation, and then list what I consider the assumptions that he makes more or less unconsciously. The reason I'm doing this first is because otherwise, if I put it right at the end, it might seem as though I'm sniping from the rear or something like that. Then I want to briefly kind of summarize the intellectual background of this, of the situation, that is, say, mostly 20th century, mostly modern, as a sort of scene-setting thing, and outline some of the people he met and the sort of frictional points that resulted. And then we can go through, in biographical order, his discoveries and rediscoveries and the reactions to them. And then perhaps at the end we can do a sort of reprise and go over them again. Okay, that's my policy.
That's quite a mouthful, yeah.
It's quite a mouthful. But it has to be done, isn't it? Okay, right. First thing is, now I want to go through what I consider to be weaknesses. It's just a fairly short thing here.
Do you want me to answer them one by one?
If you could do them at the end, maybe. It's that much. It's not a huge amount. Okay, there are 5 paragraphs I've got here. The first one is from the lay point of view. You tend to use phrases which, of course, is hardly. Well, the first one is the use of technical terms, which, although they seem explicit, in fact include assumptions. I'll give an example. To stimulate a brain cell, you're used to this, but from a lay point of view, it's hard to know whether this means applying a match or pricking with a pin or getting a battery with a couple of terminals and sticking them into the thing, and for how long, and so on. So stimulate a brain cell is a typical example. Another example is artifact. Normally this is used to mean merely just some sort of man-made object. It's rather odd to find its use in microscopy. Another example is an example he gave was using brain slices. You said that a brain slice shows the effects you get in epilepsy. Now, you're used to dealing with brain slices, but from the lay point of view, this sounds rather odd. It makes it sound as though the slice foams at the mouth or goes rigid or something, which obviously it can't do. Similarly, homogenize. Why homogenize things? When you're discussing this, you take it for granted that this is a thing that people do. It took me a long time to realize that basically the only reason for this is because cells are very tiny things, and it's a way of kind of collecting together the various types of juice. At least that's the idea of it. That's the first point. There are technical terms which have built-in assumptions which are not explained. The second one is you do tend to personify things that you like. For example, nature with a capital N and biochemistry. You can ask about biochemistry. You can't ask biochemistry. You can only ask biochemists. I think that's a weakness. Okay, on the vocabulary side of things, you're of course aware of this, and you've collected your own lists of misleading terms. For example, the latest batch being dealing with signalling and such related terms as that. And of course also verbal tricks that people use to evade questions. But you don't seem to have generalised this to the whole of medical science. I mean, for example, I remember interviewing you about malignant and invasive, which have peculiar meanings in the medical profession, not the same as ordinary meanings. Similarly, you have the idea that AIDS is complicated, while, for example, TB appears not to be complicated, and all that means is that it's curable. Okay? There are 3 things now. Another one is the business about the social effects of things, which of course is difficult to deal with. A thing that's missing very largely from your work is an assessment how actually important it is that if 60 or 70% of work is wasted, is the other part perhaps valuable? I mean, it might counterbalance the effect of this wastage. There is in any case huge wastage anyway when you think of things like education systems or whatever. Colossal wastage in some sense of the word. The final point I've got here is to do with the way you present things verbally. In my view, you should say exactly what you mean rather than what other people mean. So that if you're dealing with what you regard as a non-existent object, you should put it in quotation marks or refer to it as a so-called endoplasmic reticulum or whatever, or as a mistake or myth or something. Otherwise, the reader tends to be baffled by sudden apparent changes in view. And, for example, I mean, you do in fact do this yourself, which is sometimes, which is rather baffling. When you sum up, for example, I've got an example here, discussing molecular mechanisms of drug actions, you say, a large amount is known about what they do, but remarkably little about how they act in the living person or animal. And you present this as though it's a fact, but in fact, of course, it's just your own view. You suddenly switch from summarizing other people's views and then putting your own view to assuming your own view is correct. So these are the objections, these are what I consider to be the weaknesses in your presentation. I thought I ought to get these over with at the beginning. Do you have anything to say about all these?
Well, I have. In response to those particular points, first of all, you must appreciate that most of my publications which I've given you are addressed to professionals and not to laymen or even interested laymen. So when I talk about stimulating a brain cell or artifacts in electron or light microscopy, the concept of artifact is well known to everybody in the business. The concept of stimulating a nerve cell is very well known to people in the business. I think that you are asking me why I've addressed the wrong audience. And then I've addressed my peers. I haven't addressed the public in most cases. The only book actually I did address the public a bit was the one on the case for new paradigms, which is partly addressed to the public.
Okay, that's one of your most recent books, wasn't it?
Yes. Though the personify things, I wasn't aware of that, and it's an interesting point, but I don't think it's very important. But
okay, it's not very important.
No. Verbal tricks in language, I'm very, very much aware of that. And that's why in, I think, 3 of my books I put lists at the back of words which are simply misused. They try to make a word bigger. I mean, to take the current situation, this decommissioning of arms is now used instead of handing over of arms because they don't want to hand over arms.
That's right. You've got lists which have increased with each book.
That's right.
But the point I was making there was that you don't seem to generalize it to medical science. For example, this use of the word malignant and invasive.
No, but these are well-known technical words. I didn't invent them.
Yes, but there are reasons perhaps behind selecting particular technical words, and they can be misleading. I mean, that's the point of it.
Yes, well, as I say, bearing in mind that the books are addressed to biologists.
Yes, but you might argue that there's a technical use of vocabulary to say that decommissioning arms will be done now. Okay, that's a technical use used by interviewers, isn't it?
Well,
you wouldn't accept that, and similarly, it's legitimate to criticise the medical profession for, at least in my view, for using words which are subtly misleading. But if you don't accept that, that's fair enough. Well, I'm just making that as a point.
Yes. Now, you make a very important point about my use of either their view or my view. Both, you lose out on both of them, because if you just use their language and their views all the time, I do that sometimes to show the inconsistencies in their views. And then I usually go and say what my views are. But both of them are subject to criticism, because if I just use my views... the reason why I say that AIDS is complicated, because AIDS involves immunology and genetics. Of all the areas I've ever written about and know anything about, genetics is the one I know least about, and immunology is the second least. And therefore I'm extremely unanxious to, because I believe that if I spent a lot of time looking at immunology, I'd come to the same conclusions as I have about other biochemistry, but as I haven't, I don't want to, I don't want to get into that one.
Okay. All I was saying is that's just an example. People don't regard TB as being complicated, although that presumably also involves things like immunology.
Well, it's not mainly immunological. People think that HIV is...
even if it involves it a tiny little bit, then that's still part of the disease, isn't it? But the reason that people think that TB is not complicated is because it's curable. It's got really nothing to do with complication at all. So that's a kind of misleading use of the word complication.
Well, the theories of the genesis of AIDS are quite complicated.
But that's not the point. The point is, if it was curable, people wouldn't regard it as complicated. I mean, it's purely a linguistic point I'm making.
Yes. Well, I think that anybody is entitled to say, and I've told you many times, that this is outside my ambit of interest.
Do you have anything to say about the use of language by the medical profession? I mean, you must have watched it evolve and change.
Well, there are two totally different areas. One of them is misleading terms, which I've put back through my book. But the other is terms one uses with patients so as not to alarm them.
All right, so there's just the two things. You haven't, on the general vocabulary, the problem of the vocabulary of medicine, you have nothing much to say.
Not really, no.
Yeah, okay. So the next point is this business of how important it is. What are the social effects? If a lot of it is wasted, is it not possible they might be doing good work? Because this is what they'll say. If you say your lab is wasting 2/3 of its time, they'll say, well, this may be true, but nevertheless, the rest of it, we're doing this valuable drug addiction work or something.
Well,
and in any case, there's a lot of wastage.
Yes. Well, the answers which I've given to that are as follows. Firstly, that if 60 to 70% of all the people and the resources are wasting their time, they could actually be using those time and resources to investigate things like cancer, multiple sclerosis, schizophrenia, the important diseases, properly with appropriate technology, and they might come to much more accurate answers.
Right. Presumably there's no guarantee that they would do. I mean,
but I've stated in one of my books that I make as a reasonable assumption that the more accurate and more logical your experiments are, the more likely they are to give true results. And I think everybody does that. I mean, otherwise you just wait for a monkey to type out Shakespeare. Do you know what I mean?
Yes,
we all assume that if you make a thing more efficient.
Okay, but from their point of view, presumably the risk is that there's a leap in the dark. They don't know what they're getting for. It may be that they know what they're dealing with at the moment, even if it's wasted, whereas work in fields which they're not used to, first of all, might expose their incompetence, and secondly, might lead nowhere.
But that is I agree with you, but that's a general point of all research, isn't it, really? That you never know whether it'll lead to anything.
What about, as regards social effects, what about the argument that there's in any case huge wastage? If you look at weaponry or the amount wasted in education or the civil service or something, there's an argument that there's huge, maybe it's no worse than anything else. It may be better.
It may be better, but I mean, again, is one going to accept that people are wasting their time as a norm? If you think you can identify where they're wasting their time, isn't it your duty to point that out in order to encourage them?
Okay, but you don't have a way of comparing wastage in this particular field with other fields. You haven't done that.
No, I haven't done that. I don't think you can, probably, because they're so different. I mean.
Okay. What about this linguistic point here, which in my opinion was very forcefully borne in upon me when I was reading your book on cell structure and the mammalian nervous system, that you tend to things that you regard as non-existent ought, in my view, to be always presented as that way, because if they're not referred to as being so-called this and that or the other, it's very confusing for the reader, because it looks as at one point as though you're presenting a conventional view, and they'll think to themselves, well, what's the point of this? And later on you present an opposite point of view, and then you tend to sum up as well, so it's not only is it sort of 3 times as long as it might be, but you have apparently contradictory views throughout the passage.
Well, I
mean, that's a difficult problem.
It is a difficult problem. I think that my experience has been, if I go to any meeting, and I've been to very many, and I address it, and I tell them all the things which are artifacts, they get so emotionally upset that they don't listen to anything I say. So
in other words, you present, you tame your presentation so that first, so you try to introduce it to them.
Yes.
So like deconditioning somebody against spiders, you expose them to a little one, and then it gets bigger.
That's right, exactly. But you have to do that.
But
because
nevertheless, that is confusing. You accept that, or at least do you accept that could be confusing, because somebody dipping into a book at different points will get a different impression of what's going on.
Yes, but I mean, it is exactly a theological question. I mean, if you just said that the painting in Siena is a wrong painting, you can probably win that battle. But if you go to, say, a religious authority and take the Bible and attack it on so many different places, you'll just react in a very hostile manner and won't even listen to what you're saying.
What about the point, the claim I've made here that, say at one point, on molecular mechanisms, that a large amount is known about what drugs do, but remarkably little about how they act in the living person or animal, and you're presenting that as a fact rather, implementing your own view as a
No,
it
it isn't. Most people agree with that. I mean, you can open any pharmacology book and they'll tell you that.
Well, they use phrases like monoamine oxidase inhibitor, don't they, which suggests that they think they know quite a lot about how they act in the living person.
But that's not the mechanism of how they act. So that's the point I'm trying to make. A mechanism is different than what they do. In other words, the answer to the question, how do you do it, not what do you do.
Okay, the second small section I've got here is what I consider to be typical assumptions, which are derived from, well, largely left over from the 19th century, really. For example, your kind of Eurocentric attitude to history, Greeks, the Romans, Middle Ages, then Germany and Britain as the usual kind of picture. For example, you claim that control experiments were invented in the 19th century and that the Greeks invented scientific method. In my view, your presentation is weakened by these claims, which may or may not be true. For example, it's known that the Chinese, according to Needham, that the Chinese knew about the circulation of the blood, well before Harvey. Yes, but there's another example here about Vitruvius. Yes, that's right. Ignored for 200 years, or at least not ignored, but
Vitruvius. No, I don't.
Vitruvius. 200 years. Galen was preferred because his stuff was written. This was your claim, and I mean it must be a fairly standard claim, as with Galileo. But I'd like to know whether you actually have any evidence for it, and have you looked up what people said about Vitruvius and Galen, or you just kind of assume?
No, but I didn't deal with Vitruvius. I dealt with Galen.
Yes.
The fact was that Galen died in about the year 120.
Yes.
And when Andreas Vesalius.
Vesalius.
And Leonardo and various other people did observations on the anatomy of the human body, people denied them on account of what Galen had written.
How do you know that?
Well, at one time I read the history of it. I mean, there were several people attacked Vesalius on these grounds. Just to give one example, Galen said that there were holes between one side of the heart and the other. And when they looked and they found there weren't any, and Vesalius said there aren't any, they said, they must have closed up after the person died.
Are you sure that's not just a traditional view? I mean, how serious were these oppositions to Vesalius?
Oh, quite serious. I mean, don't forget...
How do you know?
Well, because there was a great... the church actually gave permission for anatomy to be done in Italy.
Yes, I know.
And it hadn't been done before because of the opposition of the church. But it wasn't just the church, it goes back to Egyptian times.
Yes, I know. These are these standard claims made. This is all tied up, for example, with the Reformation. I'm not saying it's not true. I'm just saying. Have you gone into it enough depth to make sure that it's true?
Well, when you ask me that question, I will plead guilty to the fact that I have depended on people who I think are authorities.
Yeah. Okay. Let me ask you another question. This is another small part. You, in my view, use the word provable in an ambiguous way, in a kind of everyday sense, which most people would use the word. But there's in the abstract kind of Popperian sense, in which nothing is provable. He relies on the idea of disproving things to kind of shave away all the errors. Do you think there's something in that? I mean, you're continually saying that things are, for example, unprovable, completely unprovable.
Well, you see, I go along with Popper insofar as he's right by saying the thing has got to be disprovable, but it's also got to be provable as well, because you've got to have some reason for believing in the first place.
Yes, but from the proper point of view, the word provable just doesn't mean anything. That anything that's left over after you've disproved.
I know, but that's what I'm saying. That's why I disagree with Popper, because if you take that view,
right, so what do you mean
don't know why you believed in the first place.
What do you mean by provable? Is it possible to state that?
Yes, you bring evidence for its existence.
Well, that doesn't...
Yes, so bring communicable evidence for the existence of something is how you prove it.
What about unprovable? If somebody says a hypothesis, for example, to do with free radicals is unprovable, what does that mean?
That means that you cannot do experiments which test the hypothesis.
Right. You don't just mean that they're extremely difficult, you mean it's actually
impossible?
No, I think
that some of them can't be done. For example, the structure of the cell membrane is supposed to be two lipids and a protein, And there's no doubt it fits in with that, but it is by its very nature not provable. It's too small to be provable.
With modern techniques?
With any techniques.
With any techniques?
It's uncertain.
How can you be sure that it's unprovable with any technique? Why couldn't you have some tiny little device, some miniaturized little engine that nibbles through a membrane and measures the distance as it goes?
Well, because you then get down to size of particles, which will be affected by what you're actually doing.
Why couldn't you get a little tiny radar set that measures it with great accuracy? I mean, I'm not suggesting that's a plausible way of doing it.
Well, if you notice in some of my publications I put unproved, and some of them I put unprovable. In my view, those ones which cannot be proved are unprovable. It may be that my knowledge isn't sufficient, or in the future there may be techniques which will permit one to.
Okay, so how do you decide what in the way you work makes you decide that something is unprovable? I mean, completely unprovable, per se for all time. Such and such a thing is unprovable.
Well,
what is it about it?
I think, for example, if you I've done it myself. We try to weigh a brain slice, you see, and as you're weighing it, it changes its weight while you're doing it because water evaporates from it.
Well, couldn't you, if you measured it in a saturated atmosphere, wouldn't that counteract?
Yes, but I mean, perhaps put it differently. If you're trying to weigh a block of ice in the tropics,
are you saying it's for all time it would be impossible to weigh a block of ice in the tropics?
Well, without having an air-conditioned room, in which case it would no longer be the tropics.
All right, let's stop at that point. Now, what I want to do here was do a bit of intellectual background and scene setting, which is, this is gathered from various sources, and it's not intended to be exhaustive or anything, right? But I think it's important to mention these things just to see what's going on, so to speak. Okay, now, I'll start with the interwar period from our point of view, between the First and Second World Wars, okay? Now, I've got a list here of things that happened. For example, there's an extension of Pasteur's work on bacterial infections, right?
Yeah.
There's a development of things like various drugs, aspirins and so on, the invention of things like penicillin, vitamins were discovered, pesticides were kind of elaborated, and parasitology and entomology and so on were evolved, weren't they? Things like TB sanatoria became obsolete or obsolescent. Viruses were identified as pathogens without really much idea of what they actually were. At the same time, there was a simultaneous idea, a political idea, that hygiene was the important thing in health.
Yes.
There were things like the Davson-Danieli thing came along in 1936, I've got here.
Yes.
There's a certain amount of dogma kind of creeping in, or perhaps it was always there. For example, around about 1940, I remember you telling me that ulcers, it was deemed really that ulcers were caused by acid and no further kind of work was done on them.
No, it wasn't just
Stop.
Yeah.
And there's a further dogmatic thing is shown in things like, for example, in the modern day, if you look at the British National Formulary for doctors, it's very standardised. There's no indication of the relative reliability of the different treatments, as though they're all presented as though they're exactly equally on equal footing. Okay, so that's the between the wars atmosphere, between the wars things. Now, after the Second World War, there was a kind of change or development or something. There was the NHS, there were things like UNESCO and the World Health Authority, and for example, oral polio vaccine was tested in 1957 to 8 in the Congo, right? Things like the contraceptive pill were invented, and also side effects, problems of drugs like cortisone became sort of notorious. That was more or less invented then. Popular microscope clubs fell out of fashion, as opposed to between the wars. Now there are only 3, including the Queckett. I remember you saying that in a lecture. Cellier more or less invented the stress theory in 5 volumes around about 1950.
Well, he invented the concept of stress.
Yeah, okay. Yeah. This sort of theory was opposed by things like the autoimmune theory of things like rheumatoid arthritis, and according to a quote here, these theories were so irreconcilable their adherents more or less ignored each other, and that was written in about 1970. Okay, and to look at technology is important. It's very hard to assess because there's a lot of backroom stuff which is hidden away. Okay, a lot of people know about things like even things like nuclear power stations and aeroplanes and cars, but very few people know what centrifuges are, or flame photometers or whatever, or electron microscopes. And so this is a very difficult thing to assess. It's not just the question of the money of them, it's also the sort of obscurity of them. And for example, you gave a list of improvements in technology, and one of them included transplantation as an example of improved empirical technique, which of course sounds different from the orthodox view by people like Medawar that immunology was an important science. Mm-hmm. Anyway, so the present situation is, roughly speaking, that cancers, degenerative diseases, so-called, of heart and its arteries and so on, multiple sclerosis, Alzheimer's disease, and things like schizophrenia and manic depressive illnesses are, well, not just incurable, they're not understood. Moreover, about half the top 10 of the NHS operations are regarded as being of dubious value. Right. Okay, so that's all right as a vague outline.
Yeah.
Okay. Now, the next thing I've got here is, this is not quite to do with the intellectual background, it's to do with your personal background. There's a huge list of people in your writings. There's also a huge list of people who are not, whose names you have not mentioned.
Yes.
Broadly, I mean, these are some that I've come across. I'll read out what, for want of a better word, I'll call baddies. There's an extensive list we have here. De Robertis, who wrote about the synapses, yes. Dr. Eales at Guildford, who won't talk to you.
Dr. Eales?
Eales.
Eales?
It's just possible that she's busy. Well, I count her as a baddie for not turning up. I mean, she presumably had plenty of opportunity to speak to you.
Oh, yes.
Well then. Okay, Porter you didn't like, and Walpert. I think at one stage you said that neither of them had done anything in their lives or something.
No. Porter is different. Porter invented the microtrabeculae. And he was a man at the International Congress of Cell Biology in Berlin in about 1981. I asked him how it was possible that there should be a cytoskeleton while there were intracellular movements, and he wouldn't answer it.
Right. Okay, Walpert perhaps was the one that you regarded as being a windbag.
Yes, but I'd rather not quote him. I mean,
okay.
There's a personal opinion, it's not scientific.
Yes, that's right. McIlwain, who I think you said you wouldn't publish anything against his own views.
Yes.
There's Houdin. Now, I think perhaps he was the chap who crossed out the results. I don't know, somebody did. There's Berenbloom, Huxley, Cummins, Chan Pillay, who attacked you recently in Singapore, B. Chance, Bernard Katz, Prof. J. Lucy, a huge batch of people after your school science paper, including Horn, Harris, Michel, Finian and Coleman.
Well, those are the only two published papers.
Yeah. And who else is there? There's Jennings promoting his EEGs. I think there are people promoting the neutron diet as well. I just read about that in Which recently. Also people like Talcott Parsons, you didn't think much of.
No.
Indian and Chinese people cheating with their flame photometer computer outputs at Bangor's Schizophrenia Association while it existed.
Well, I'd rather you didn't put that. I don't want that to be reported. Okay. These are personal things I told you, and I wouldn't like those to get out. I mean,
all right.
Okay. They're not relevant to science.
And who else is there? There's an anonymous head of biochemistry at Surrey who had a butter yellow theory, which he got all wrong apparently. Electron microscopists like Lieberman, Brightman of Washington, and Geary at Charing Cross, who claimed to have microphotographs,
yes, but
over many years failed to produce them.
Yes.
Susan Greenfield is another example of someone who appeared not to be willing to discuss your papers.
Yes. So
these are sort of baddies, roughly speaking. There's some cowardly ones as well, you might, for want of a better word, like McKean, who visited, but apart from putting a little paragraph in his school biology textbook, have gone along with the establishment view. McKean of O-level biology book.
I don't think you know. It was Roberts who put something in his A-level biology.
Oh, right, Roberts. And you've got a tiny list of goodies, including your collaborators like Dave Jarman, Ernest Chain, who seemed to find your work, when everybody else was saying this should be suppressed, said it was respectable. Olaf Lippold, who apparently was more or less squeezed out for his EEG theta wave and eye movements theory. Barbara Banks, publishing on ATP, and David Horobin.
Yes.
So those are the sort of personal things.
Yeah, but I would like to distinguish between my scientific life and my personal life. I mean, my view of Walpert is absolutely personal. I told you privately I wouldn't like anybody to know about it.
Oh, really?
Because it's based on a personal friction I have with him. It's not based on scientific argument we've had over, except that, I must say, he was the editor of a journal which didn't treat me fairly.
Which journal was that?
Journal of Theoretical Biology.
You mean he wouldn't publish your papers or something,
yes,
or letters?
And he wouldn't publish my papers, and the reasons he gave were quite extraordinary.
Okay, well that's fair enough, isn't it? Now, it did occur to me that possibly a result of this is reluctance to get involved with other people. For example, I've got a huge list here, there's a list of people like Maurice Papworth and Bryce Smith and Michael Joseph the lawyer, who all, well, some of them actually died before we got around to. I think it's rather a shame.
Well, I don't think one can generalise. I mean, if you take, just read out that list again. The first one was Papworth. Well, I was in touch with him, and I did agree to go round to him, and I wanted to.
But only after 25 years after his book was published.
No, I didn't see his book originally. I was in contact with him. He wrote me a letter, actually.
Right, yes, I know.
As a consequence of one of my publications. I knew of his work, but I didn't know the extent of it until he sent me them.
Well, and I mean, Bryce Smith, although not a biochemist, was a distinguished, and still is, I suppose, a distinguished critic of pollution and so on chemical grounds.
That's right. Well, he put forward the theory that lead was responsible for some mental disease.
Yes. Lead in petrol.
Yes.
Well, environmental.
Yes.
Well, anyway, that's just an example.
Yes.
I mean, Michael Joseph the lawyer is another one. There are AIDS people that might have been talked to, I'd have thought, and also this BSE chap, for example.
I have been in touch with Dewsbury.
You have? Oh, I didn't know that.
Yes. But as I say, I decided consciously not to get into the AIDS business because it dealt with an area which I wasn't sure of.
Okay. Right, well, that's basically all that stuff really is scene setting. What I want to do now is go through your life sequentially, and I've got a list here I've kind of put together a list including papers, books and events, including other people's papers and books, so as to get a kind of overview so far as it's possible. It's very difficult, of course, because there's so much going on. But should we do with that then? Okay, well, let's start in 1930. You were born in London. It's a good starting point, isn't it? And on the 16th of 8, I've got down here. And you went to UCS, I've got down here with both brothers, is that true? I don't know.
Yes.
Right, did you all get scholarships?
No, I didn't get a scholarship.
Okay, so anyway, in 1939, war started out. In 1941, you were evacuated for a bit, or something like that. Okay, not worry too much about that. Okay, I've got some events here of the 40s. 1940, Abbé Louyer's cryobiology microphotographs were published. 1945, things like the atom bomb and death camps were discovered or occurred. 1946, the NHS Act was passed.
Yes, but the NHS only started in 48.
Right. 1947, coal mining was nationalised, wasn't it?
Yes.
And in 1947, work published in 1948, the cat's head was isolated, perfused with liquid, appropriate liquid type.
48? Who was that?
I don't know.
I think
You've got it down. It's in one of your bibliographies. That's the cat. It's not the monkey.
It might have been Smythe.
Yeah, I don't know.
Yeah.
In 1948, the first post-war Labour government refuses to take part in sections against South Africa. Got that down.
Yes.
Right now, 1950. You were now about 20. You finished your BSc in about 1950. I've got it down here. And
No. I finished my BSc in 1958. I took my medical degree first.
Oh, did you? Ah, that's expensive. So up to 1956, you were doing an MB at Middlesex, is that right?
Yes, that's right.
I've got down here as 50 to 56, maybe that's wrong.
No, that's correct.
All right.
56 to 58 is when I took my degree in physiology. I went back to study.
Oh, I see, right. Okay. Presumably you were exposed to all the usual stuff in training. The only information I remember talking to you about it is that you were exposed to a bit of hypnosis. A 12-year-old girl was hypnotised to make her go to sleep. And you saw a BBC woman executive regress to infancy.
Yes, there's a
must have been a sight-worth thing.
A 12-year-old, it was a 19-year-old girl.
Was it?
It was in the RAF, actually, when I did my national service.
Oh, that's a different no, there were 3 hypnosis things in that case. There was a 12-year-old one you made go to sleep.
Oh, that was when I was qualified.
Oh, right. There was a 19-year-old one who was told to buy stamps next day.
That's right, that was
and there was anyway, that's just, it's not very important. And presumably you also got involved with non-invasive neurological examination of patients with brain injury. In other words, trying to work out whether...
All doctors do, I mean.
Right. Okay, in 1955 you started doing some work on resuscitation, according to what I've got down here.
Yes.
That means was that part of the normal MB, or was that a special kind of side issue?
Actually, it was 1956, not 55.
Was it?
Yes. It was when I was a houseman.
Does that mean it was one of your interests, or did they all do that?
Yes, it was one of my interests.
Okay. You also, about this time also, you met an Algerian believer in the Quran, and they were brainwashing scares. I'm just putting that in.
Well, what it was I was sitting in a cafe in Algeria, and this man told me that the whole world would be a lot better place if everybody was a religious Muslim and believed in the Quran.
Yeah, quite right too. Okay, now 56, 58, you did a degree in physiology.
Yes.
Right, and then 58 you started, this is when you were now 28, started your PhD thesis on brain slices. I think it was mice. In fact, there's a copy of it over there, isn't there?
Guinea pigs.
Was it guinea pigs?
Yeah.
Copy there, blue-bound
Yes.
thing in what now looks like extremely antiquated carbon-papered typewriting with sketches
Yes.
pasted in, that kind of thing. This time, academics were protected by tenure. Right.
I wasn't an academic, I was a research student.
Right. Anyway, first job, 1958. This was McIlwain, Prof. Henry McIlwain.
Yes.
And various things happened which you didn't like. For example, your technique of cutting brain slices was sort of hijacked and written up as a paper, and your part in it was underplayed.
Yes, well,
Right.
I varied the technique. There's a very well-established technique, and I think I improved it. And as a consequence, the boss published a paper without me on it, although it was my technique.
Moreover, there was a mistake in the paper.
Yes, it was a bad mistake.
And when you wrote subsequently, you had some trouble publishing the correct value.
Yes, correct.
So you drew a few conclusions from this, including that doctoral students haven't redress against their supervisors unless they want to lose their jobs, and that well-known academics find it easy to publish, especially if they're on the editorial board.
Yes, that's absolutely
And there's an inertia in the data reported in books, which encourages people to find similar results. That's right, I'm sure that's true. I remember when I was doing A-level, the absolute zero is minus 240, isn't it, or something? And I managed to get a value that wasn't on the line, and this teacher crossed it out,
Yeah.
I mean, that's somewhat a minor example. Okay, 1960, you're now 30. Peter Medawar gets a Nobel Prize. Next year, Amnesty International starts, and also Robert White isolates a naked monkey brain with an eye and an ear attached to it. Okay, now 1962, 32, you go to Sweden and Gothenburg, or Göteborg, however they pronounce it. And you work with Prof. Holger Huden, spelled Hyden, at the Institute of Neurology, and he's developed this technique. Neurobiology, I'm sorry. And he's developed this technique which to a layman seems perfectly obvious, of opening up a rabbit's head and taking out nerve cells with a sort of bit of wire.
Yes.
Seems fairly obvious, but apparently that's a major breakthrough. And you notice this business about people crossing out results and so on and so forth.
Yes.
And, well, here we are, not discussing results which disagree with one own, avoiding doing crucial control experiments, answering important questions with artful circumlocutions, and sort of that sort of thing. In fact, Arthur Koestler was there as well. That's that around about that time.
Arthur Koestler wasn't there. He left a manuscript with Huden.
He left a manuscript, oh right.
With Huden, for Huden to read.
And also, what else happened in 1962? Dr. Joseph Cummins from America came there.
Yeah.
He worked out a way of measuring in single nerve cell bodies the activity of ATPase, which breaks down ATP. Now the first thing really that happens to you, the first intellectual thing, what I've put down here as a discovery, the first discovery is to do with phosphate bonds, ATP and so on.
Yes.
The terminal phosphate bond is a transducer in 1964. Now.
Yeah. No.
Okay, the terminal phosphate bonds are transducer. This was the effect. You did lots of controlled experiments on things like sound and light and the effect on ATP. And these results were not popular. And they were, in particular, at a meeting of the International Union of Biochemistry in Washington. People made sarcastic comments. And also there was a meeting of the Physiological Society in Mill Hill.
Where it was rejected for publication.
Yeah, it was rejected for publication, and also people said nasty things, presumably.
Yes.
So could you just summarise the story of that?
Well, I had done about 330 experiments on the so-called high-energy phosphates, that is ATP phosphocreatine and phosphoarginine. Phosphoarginine is the high-energy phosphate of insects. And I'd done a very large number of experiments. I wrote them up for publication, and I thought that if I submitted all the data and the experiments for publication, they would be easily published. And I had the most terrible trouble. I couldn't get it published. But people said to me, well, if you give it at a Physiological Society meeting and they approve its publication of a short abstract, then it'll make the bigger paper published. But the big boys at the Physiological Society didn't like it, and they behaved quite badly about it.
Yes. Okay, you said at some point, you wrote at some point that you never quite worked out what the basis of objection was, though.
No, I didn't. I'm afraid to say I think that it was the reaction of a powerful society when a young man of 32 finds something that's quite important. This is a personal view. I have no other way of assessing it.
Why was it important? What was the importance of it?
Because phosphocreatine, ATP and phosphoarginine are ubiquitous in the body, and it, if I am right, is quite important.
Okay, in other words, you found a biochemical which is found widely distributed in tissues, and it has properties, physical properties, which must presumably have some importance, or might well have some importance.
Yes, yes.
It's like discovering that common salt, for example, has electrical and physical properties. It should have.
That's right.
Yeah.
That's right. Yes.
Without actually saying what they are, it might have implications.
Yes.
Yeah. Okay. Can I just ask this question about Barbara Banks and her demolishing the whole idea of high-energy phosphates? Does that
Yes.
what's the relevance of that here?
Well, Dr. Barbara Banks was in a totally analogous situation to mine. She had shown on theoretical grounds that the concept of high-energy phosphate, which Fritz Lippmann had got the Nobel Prize, I think, during the war in about 1940, was a non-concept from a thermodynamic point of view. Very briefly, what she showed was that the concept of a high-energy phosphate means that compound liberates a lot of energy, and it is not a compound, it's the actual reaction which it undergoes which liberates the energy, and if you put it in different circumstances, it will liberate different amounts of energy.
Yeah, I think I see what you mean. It's like talking about high-energy magnesium and then setting light to it or something.
That's right.
Vaguely analogous.
Yeah.
But that doesn't affect your views.
No. She's just in an analogous situation in that she found the greatest difficulty in getting it published, and her own job was threatened. But she didn't lose it.
I mean, I just noticed that you used the expression yourself, high-energy phosphate, earlier on.
Yes, I use the expression because it's a popular expression, but I think you'll find I put always in inverted commas.
Oh, I see. Okay, so it's spoken in inverted commas.
Yeah.
Okay, let's see what else happened here now. 1963, Andrew Huxley gets a Nobel Prize. I'm not sure what for. The Medical Research Council Unit of Applied Neurobiology.
Yes,
it's newly formed.
Yes, I was a deputy. I was the second in command there.
Okay. So you're about in your mid-30s now. All this is going on. What else happened? You worked on ATPa's activity with Houdin. That sounds as though it was what he was doing before, wasn't it?
Yes.
Okay. And you wrote a paper, Do Oscillations in Biological Systems Originate in Their Substrates and Nucleotides? Question mark, I'm not sure. What is the significance of that?
Well, you see, this was part of the attempt to get published this work on ATP and phosphocreatine. Various people have found that organisms, whole organisms, had oscillations in them, you see. I had shown that ATP oscillates. So I tried to ask those people who've shown the whole organism whether they tested the ATP, and they didn't answer.
By oscillation you mean as in daily rhythms, that kind of thing?
Yes.
Of everything?
Yes. When you shine light on ATP, and it is a very peculiar property, instead of just breaking down, it goes through cycles of instability.
Really?
Yes.
Right. Okay. Anyway, let me see what else happened here. In 1967, Maurice Papworth published his Human Guinea Pigs book.
Yes, I didn't know about it at the time.
Right. And also at the same time, George Porter got a Nobel Prize, I think it was his first one, for something to do with photochemistry, which of course is related to what you were doing.
George Porter, does he?
Yep. Well, according to a reference book I've got.
I didn't know that.
Okay, 1967, what else happened? You met Sartori around about this time, yes, 1967 you met Sartori, and he was ill with lung disease.
Yeah.
But he kept on for a number of years after that. Right, you developed this technique of taking out cells in saline. You say, which was more natural, it makes them stick to the slide as well, doesn't it, or something?
Well, yes, it shows a new membrane. Which we
right,
found.
1967, discovery, well, this was rediscovery, strictly, isn't it? Number one. Your rediscovery was, you saw a membrane around the nucleolus.
No, that wasn't a rediscovery, that was a new discovery.
A new one?
Mm.
Have I got this wrong? I thought that if you look in some of the photos that you've got reproduced in the living cell, the nucleolus is...
Yes, what people didn't recognise as a membrane, but in the actual drawing you see one.
Oh, I see. Oh, I see what you mean. In other words, you
identified
it.
We said there was a nucleolar membrane, and then when we started looking in the literature, we found there were drawings, but nobody mentioned there was a membrane there.
Right. So it's a sort of discovery which had been made before without people noticing it.
Yes.
Not sure if there's a word for that. Okay. Now, and this was not published until 1974. That's 7 years later, as a nucleolar membrane in neurons.
Yes.
You also published it in your 1991 atlas.
Yes.
Of the cellular structure of the human nervous system. Tried to publish it, not successfully. It took 7 years. 1968, the Unity Lab was founded. So you're now getting up to 40 by the Handicapped Children's Aid Committee of London, which financed it until 1981. Did you work in the Unity? I haven't got a note here. Did you work for them from then on?
No, I didn't work for them. I worked for the university, but they subsidised my laboratory. I had 1 or 2 people working for me, either doing PhDs or postdoctoral, all this time paid for by them.
Was that the Unity Lab?
Yes.
Right, so you were, while you had research assistants, the Unity Lab was founded and you moved into it.
Well, they just called my laboratory the Unity Lab.
They moved the label Unity.
Yes.
Right, now, 1970. Oh, well, this is where, yes, ATP was regarded as a high-energy phosphate, and then Dr. Barbara Banks, who had great difficulty publishing it, published it in the School Science Review.
Yes.
Is that a rather insignificant publication? Or is that not a fair thing to say?
Yeah, well, it's not, I wouldn't say it's insignificant. It's school teachers, you see. So it's very influential.
But it's not Nobel Prize stuff.
No, but it's not a central journal like Nature, which one would like to get it in, because it's a very important question.
Right. Now, meanwhile, in 1970, Bernard Katz, who made a lecture the year before, gets a Nobel Prize for physiology, stroke medicine. I don't think they distinguish them, which must have been, I presume, on what are called neural transmitters.
Yeah, theory of transmission, yes.
So that started another red herring in your view.
Yes.
Okay, 1971. Now, another discovery here. 1971, most of the new structures in cells, that is to say, newly discovered ones,
by the electron microscope,
are artifacts.
Mm-hmm.
Okay, I've taken the 1971 date from your case for new paradigms. Okay, 1967 says here, this is you and Sartori suddenly noticed that the endoplasmic reticulum, which is a network believed to be a structure in the cytoplasm, brackets the cell sap, appeared to be cut perpendicular to the section far too often. ... This was also true for other membranes like other apparent membranes and the Golgi apparatus, mitochondria. About 1971, discovery that most of the new structures in cells apparent by electron microscopy are artifacts, new structures being ones supposedly discovered after optical microscopy. Okay, there was the cuts perpendicular to the section argument involving Golgi apparatus, mitochondrial membrane, cell membrane and nuclear membrane, as well as the so-called endoplasmic reticulum. Right. Okay, there's the perpendicular argument. There's also the argument that if they existed, intracellular movement would be very difficult, if not impossible. So you collected overwhelming evidence. You submitted a paper. Nature rejected it. The referee said that at that time, in 1975 it says here, in other words, it seems to have taken 4 years to get a rejection. Is that right? Still believed in the unit membrane?
No, we sent it to all sorts of people, but Nature said that they agreed with it.
Did they? It says here, the referee believed that no one at that time, brackets 1975, took that from your intellectual recovery. That's right, is it?
That's correct.
So the thing was still going on 4 years later.
Oh yes, I mean we started trying to publish in about 1971, I think.
It says here that the discoveries with you and Sartori were doing this work in 71,
Yes,
thumbing through electron micrographs and not being happy about them.
Yes, sir.
Science gave no reason for rejection, and so on and so on. And cytologists said you don't want to believe what you read in textbooks,
Yes.
which led you to write a 1977 paper, Value of Textbooks. A letter, right.
It was a letter to Nature.
And you also put a list of books, partly because there's a list of books as a bibliography to the Living Cell, 1918, in which all these beliefs were explicit. I forget how many, 30 or 40.
Yes, what I did
textbooks.
is I got there are some reserved textbooks which you aren't allowed to take out of the library, which are the latest editions.
Right.
And I got all of them, and every single one of them said what Nature denied was commonly believed.
And that was 5 years later, 5 years after the chap in Nature said it, or 4 or 5 years, and then you put it in the bibliography of the Living Cell, which was 1980. But that's in the future. That's still 10 years ahead. You're now about 42, 1972. Now your next discovery was that at least some, if not all, popular, important biochemical research techniques haven't been controlled, and you published this as 1972 Certainty and Uncertainty in Biochemical Techniques. And incidentally, this year was the same year that Singer and Nicholson published their fluid mosaic model of the cell membrane.
Yes.
Okay, now, well, you got interested in this. You started with electron microscopy, and you started with fractionation, and subcellular fractionation as a result of working with Houdin.
Yes.
Then you investigated electron microscopy. I think that was the order, and then histochemistry. And you also did 3 other ones, which were sort of less important.
No, they're very important. And altogether, the 6 of them are the 6 most important techniques used in biochemistry all over the world.
Right. So anyway, there were 3 which you started with, and then there were 3 techniques nearer the measurement end. You put them chromatography,
that's right,
electrophoresis, and radioactive measurements.
Yes.
Each had their burden of assumptions, and you sought permission to do control experiments, and was told they would be a waste of time or controversial. You wouldn't be able to get the results published or whatever. The SRC, the MRC, and the University of Surrey wouldn't support them. None of these would support them.
Yeah.
And hence you wrote your book, of which not a huge number were printed. I mean, 2,500 copies.
No, that was quite good for a book. 2,500. It was quite good actually for a science book.
Not by my standards.
No. And also, I told you, 12,500 was sold out in Russia.
So they say. Yes, anyway, it was reviewed by Nature. You've got various reviews. The Times Higher Ed supplement, Science Progress, Acta Biologica Academica Scientia Hungarica. And basically, I think, your methodology in this book was to take these 6 methods. Now, from the layman's point of view, what was a bit worrying was that you don't give, as I explained at the beginning, you didn't give reasons for why these things were done at all. I mean, for example, it took me ages to work out that subcellular fractionation was done simply because these things are tiny, and therefore you collect them together. So it does make some sense, prima facie. Similarly, with electron microscopy, it's got huge magnification, so it's not surprising people should try using it. Okay.
But again, bear in mind that I am actually addressing professionals who would know these things.
Yeah. Anyway, your methodology basically was to compile long list. I mean, I suppose long list is being unfair. 8, 10 or so objections. You try to list one by one methodologically, which is a very difficult thing to do, of course, isn't it? Because there are all kinds of unconscious assumptions.
Yes.
It's like trying to list what you do when you make breakfast or something.
Yes.
There's a whole lot of things.
No, I agree, but my main contribution to thinking at that time, I believe, was to list the assumptions which people hadn't even recognized in the techniques that they used. And these assumptions were inevitable, inescapable.
Right. Did you distinguish the inevitable ones from the less evitable ones?
They're all inevitable.
Right.
Because some of them, as I showed, were contrary to laws of physics or thermodynamics.
Yeah. Just that 2 of the reviewers, there is a kind of methodological point which I could see their point of. They said, I mean, for example, this Professor Lucy of the Royal Free Medical School said that you'd overstated your case in stating that the validity of a localization of an enzyme activity, whatever it is dependent on all 15 assumptions being warranted. And then he added it's sick. Now, I mean, I can see a point that said if you just get one crucial objection, then in a way that's sufficient to demolish the whole case, isn't it? If you've got a huge list of them, it's kind of adding on.
No, but it makes the case more powerful.
Yeah.
But it's also true, as I put somewhere else, that the strength of a chain depends on its weakest link. And if there's one weak link, the chain breaks. And it's the same with an experiment. If you've got a whole... in fact, in those experiments, there were 22 separate assumptions.
But you didn't list weak links in particular. You listed...
No, I listed
all of the assumptions on the same footing.
No, I listed all the assumptions, and the reason I did that was because I knew that if I listed only those ones which were wrong, which were unwarranted, they'd say I was attacking the technique. So I listed those ones which were warranted and the ones which were unwarranted.
Yes. Okay, now, I've got some notes I made when I read this book. Harold looks at 6 techniques by which the biochemistry of biological things is looked at. Exasperatingly, he doesn't survey the entire field of techniques, so he can't tell how important these are, nor does he indicate their relative importance, or even the history of development, so that we can at least see how they developed. For example, the sort of techniques that strike a layman are things like growing things on petri dishes, the use of X-rays, just ordinary optical microscopy, things like LD50 tests on animals, injecting things, breeding and growing plants.
Let me tell you that
empirical testing of pharmacological substances, physiological testing of exercises and breathing, ECGs, EEGs.
No, the point is that I have, although as a senior physiologist, I have a PhD in biochemistry. You see, and I was addressing the 6, what I judge as the 6 most popular techniques used in biochemistry.
Yes.
Now, other people might judge that others weren't as popular, but everybody agree they're very important ones.
Do they?
Oh yes, everybody.
How important?
Well, you open any textbook in the world and ask what happens in the nucleus. In order to answer that question, they've by subcellular fractionation, they've separated a nuclear fraction.
Okay. Do you see the point I'm making? What I'm saying is that as a relatively lay person, it's like opening a book criticising, say, maths, and saying contour integration has these weaknesses, without ever saying where it came from.
Yes, but as I say, if I may say, you're still looking as a layman, and it was actually addressed to professionals.
Nevertheless, you didn't...
I don't think you have to tell a professional why subcellular fractionation is done, because he knows.
Really?
He'd be insulted if you told him.
Would he?
Yes.
Okay. Right, let me see, chapters 1 to 6, details step by step. And you also, of course, include... the chapters are not strictly compartmentalised. If you've got an assumption already mentioned, you refer to it later and so on.
Yes, just to make the book shorter.
And you have references after each chapter.
Yes.
You put quite a lot of stress on heat generation. Is that a fair statement? The whole study arose out of thoughts about the relevance of the experiments on friction carried out by Rumford in the 1790s. What it says at the beginning, I don't know, but I presume that...
There's a very well-known experiment. What Rumford did was he showed that caloric didn't exist. You know those experiments?
He bored cannon.
He bored cannon, you see, and up to that time everybody thought that each object had a certain amount of heat. And what Rumford showed was that the more you go on boring it, the more heat it generates. So there wasn't a fixed amount of heat, it depended how much work you did on it. And that was one of the most important experiments in history, actually.
So,
thermodynamics.
Okay, and you investigated thermodynamically, but of course heat was not your main interest.
No, but heat causes rise of temperature.
Yeah.
Or it causes free energy, and rise of temperature and free energy both drive all the biochemical reactions we know about.
Right. Now I've got a list here. This the last 6 chapters is actually characteristic of your books. They vary wildly in length of chapter, so you can't. The contents table is kind of misleading. Let me see, 7, 8, 9. 7 briefly summarises what needs to be known to find things out. Yes, hiatuses in our knowledge. Chapter 8 looks at the individual techniques. Chapter 9, the definition of biochemistry. Let me see. Okay, so, and Chapter 9, the chapter title is the definition of biochemistry, which has a whole lot of things in it, including looking at the word biochemistry and different uses of it, the motivations for measuring biological constituents, the actual motivations for measuring things, the procedures that make in vivo correspondence unlikely, distinction between biochemistry of leather and skin is the example you give here, and the distinction between statements like particles that one separates in a fraction can carry out oxidative phosphorylation as defined, or that the particles actually can do it in vivo, they can have this biochemical activity in vivo. And let me see, comment on heat, on control experiments, And okay, now can I just ask a question about uncertainty? I wondered whether it did occur to me several times whether the so-called uncertainty principle of the 30s was kind of surfacing at some points here. Is that influenced
Well,
you?
but yes, by analogy it was. In fact, I think I told you I wrote to Heisenberg and asked him if he'd write the introduction.
Oh, really?
And he said, as he wasn't a biochemist, he didn't want to.
You're still alive then?
Yes.
No, I didn't know that.
But it's meant to be an analogy. That is to say, that in order to look at enzymes, you have to mash up the cells, and therefore you can't mash up the cells without looking at the effects of mashing itself.
Oh, right. So it's a it's not just uncertainty in the sense of things being hard to measure. It's actually analogous to the idea of the electron being hard to view.
Yes.
Exactly, and it's pretty much analogous.
Yes, it's analogous, but it's not the same thing.
But I mean, it's very strictly analogous in the sense that the actions of the operator are influencing the results.
Right.
Yeah, okay, fair enough. Okay, chapter 10, practical conclusions, very short chapter, which says what you've already said, plus comments on emotive words and evasiveness. And this is your first appendix 1 here, is your first table of kind of fraudulent words. Yes. Chapter 11 seems to map out the proposed study of what should be done. And then there's chapter 12 as well. And there's also a couple of mathematical appendices contributed by somebody else on thermodynamics of rapidly rotating tubes. Is that a fair statement? Is there anything that you ought to be added to that?
No. I mean, I put the problem to a mathematician, and he worked it out for me.
Okay, you didn't do mathematical analysis of any other techniques?
No, I couldn't.
I don't know whether it's possible to.
No, you can't.
Right.
There are too many unknowns in them.
Okay. Well, what else happened in 72? The ILP allowed Labour Party members in. I've got down here. And you also started publishing articles in I think it was a nurses' magazine on architecture, town planning, air pollution and disease.
It was a journal called Hospital Update for doctors. It was a nurses' journal. It was a hospital update. I did a series on human biology for about 12 articles on things like air pollution and sewage and water and violence and
war and civil disturbances.
Yes.
Okay, 1974. You're now mid 40s. Your nucleolar membrane in neurons is published in Microscopy magazine. That's the Queckett journal, is it?
Yes.
So that's a fairly minor.
Yeah, well, it's got a circulation of 700, but it is not a central one. I must say, I tried to get it in Nature, and again I had a huge battle. And I offered to do what the old-fashioned thing was. I used to do when Nature was first started. If anybody discovered anything, they'd actually send it to the journal. So I sent them a slide showing the nucleolar membrane. And I said, "Would you send this to the referee for them to look down the microscope and look at it?" I also sent photographs, of course, and they wouldn't put it in, but eventually I got it in the Queckett.
Do you know if the microscopist ever looked at it? Did anybody?
Well, I had a most extraordinary experience. I went with Sartori to Oxford at the Physiological Society meeting, and we showed a number of people, and they looked down the microscope and they said they couldn't see it. But one of the greatest microscopists of all times had been a man called John Baker. He looked down and he agreed he had seen it, and we were very pleased about that, because I'd asked him to come along and look at it.
I see, a bit like Galileo. Not.
I mean, I can show you. They actually don't believe it's there.
Okay. Now, what else happened? Let's see. 1975. Prof. Andrew Willie, W-Y-L-L-I-E, invents the idea of apoptosis,
yes,
which you maintain is unproven.
Yes.
Or perhaps you maintain something stronger than that. I don't know.
Well, I'd rather not go farther than that at the moment.
Okay, 1976. What happened here? Claude Dedouve and Pallard together got a Nobel Prize for work on subcellular fractionation and its relation to electron microscopy. In other words, presumably what they did was get a whole lot of stuff, fractionate it, bring it under electron microscopy, and discover all things like the electron.
In my book, I made a mistake,
reticulum,
which a reviewer pointed out. His name wasn't Claude Dedouve; it's Christian Dedouve. And a review of my book actually.
Yeah, there are three people on it: Claude, Dedouve, and Pallard.
Oh, that's right. Yes, that's right. But his name is Christian Dedouve. I put his wrong.
Oh, right. So that was your major error, was it?
Yes.
It's very important, of course. And also Slater, S-L-E-Y-T-R.
Yes, and Robards.
Robards invented freeze fracture, so-called.
No, they didn't invent freeze fracture. What they did
wrote about it.
They wrote 3 papers showing all the artifacts in it. And having shown the artifacts, they ignored them all in their own work, which I think is quite extraordinary. And they also published papers on the freeze fracture of water, you see. And all in water you can see all the membranes and all the things which they claim are in living cells, which is quite extraordinary.
Okay, you mean, for example, the crystalline things with parallel lines, all this sort of stuff?
Yeah.
Well, in fact, okay, what else happened to it? Black and white film was made. 70s, you were in your mid-30s. No, 46 you are. 46, yes. Black and white film made with the audiovisual department of the University of Surrey. Now, this must have been a film, mustn't it? It's pre-video stuff, called The Fine Structure of the Living Cell, a reconsideration, with yourself somewhat younger than you appear now, doing the sort of voiceover. Okay, black and white film, it was about, as I remember, it was about half an hour, wasn't it?
Yes.
You also published Mechanisms of Drug Action, a New Approach, and Value of Textbooks. You also got into the Observer, actually, letter in the Observer, because by this time, Sartori in particular wanted to widen the debate.
What had happened was that what you missed out there was that we actually published our first important paper saying that the endoplasmic reticulum was an artifact.
Which paper was that?
It's called something like that.
The endoplasmic reticulum is an artifact.
Something like that. In 1977, Sartori and I published a paper in the journal Perception entitled "The Unit Membrane, the Endoplasmic Reticulum and the Nuclear Pores Are Artifacts."
All right, it was in Perception. That was a psychology magazine. I remember you telling the story of that. It was a psychology magazine which wouldn't normally have had such things in, so you had to rewrite it slightly with a geometrical slant.
Yes.
Right, is that where you introduced the, your onion skin kind of drawings?
Yes.
Yeah.
And when that came out, it appeared in the Observer of 1977.
Well, I've got December 76 as a letter in the Observer.
Oh, it's December 76.
Well, that's what it says here. I don't know how.
Oh, that's right.
Yes,
it was December 76. And then we were strongly attacked by the officers of the Royal Microscopical Society.
Who was that?
Who?
Parrish. All of them, I see.
Yes.
Was that somebody called Parrish? I remember reading it.
Oh no, Parrish reviewed my paper. No.
Not him.
It was Everett, who's still the secretary of the Royal Microscopical Society, Robards, I think it was, Finian and Coleman, I believe.
Okay, I've got a gap here. Okay, 1980 was when The Living Cell was finally published, but I've got a sort of gap after about 76. You've got Duduve and all these people and your black and white film. Incidentally, in 1979, Horobin discovered that Valium activates cancer.
Yes.
79, according to Martindale's pharmacopea, and that must have been when he got sort of thrown out soon after. Is that right? Is it a fair comment?
It sounds right, yes.
And you also wrote in 79 to the British Biochemistry Society Bulletin asking for references on controlled experiments. And
that's correct, I
no
did.
reply. There was also this thing in Thuringia, the Leopoldina Symposium, whatever it is, on cell
they
structure.
wouldn't publish my work.
Also, he also wrote to Medawar, enclosing a 4-page summary, and he referred you to his electron microscopist who seemed to have been Dmytrovsky or something.
Yes.
Is that right? And it says 10 years later we haven't heard from his man, who would now be 15 years old. There was also a recently knighted Oxford
Can you
professor.
cross out Dmytrovsky, because I can't remember his name.
Okay,
it was
not.
His name.
I remember I asked you that. We were going through a book by Smith's book on viruses, about 1963 or something, and his name suggested to you that was the same chap, but you weren't sure then either. Right? Okay. And also, a famous textbook writer agreed that the Robertson model of the unit membrane was impossible.
Yes.
And said he couldn't take it out of the next edition. When I asked him why not, he smiled benignly.
This is correct. I mean, I could tell you his name is very, very well known.
But you're not going to tell me his name?
All right, I'll tell you his name. His name is
If you can remember
Dr. Hugh Davson.
Davson,
Yes.
of the famous membrane.
Yes.
Oh, I see. I didn't realise. I don't know. I assumed he was dead or something. Okay, then in 1980, the living cell. Oh, there was a debate also with. Oh, this was after the living cell. Now, so the living cell it took some time to get it published, and it was published by Packard, fairly
which is a small publishing.
It's a small. They've done quite a good job of it.
Oh, it's a beautiful job. Yeah.
What happened in those 4 years or so? Okay, there's a nuclear membrane about 74, and a mechanism of drug action about 76. The approach. So there's about 3 or 4 years there where you were doing work. So it was before you started working on the brain, and you were all this time you were working on this same business, were you?
Yes.
So that was looking at artifacts going through electron micrographs.
Yes, and I mean answering a huge correspondence, and giving lectures all over the place. I've got a list of the lectures I gave. So I give lectures at least 150 universities and institutes.
In which countries?
Britain, United States, France, Germany, Sweden, Denmark.
Was this in English?
Yes.
And the results, I didn't get that impression from your book. I mean, I suppose it's partly because it's difficult to document, and so that when you give your accounts of people meeting you afterwards and saying that they agree with you in private, but won't dare, don't want to prejudice their careers. That's how many people was that in total? And did it happen with every lecture?
Nearly everyone.
So there must have been allowing for multiple people at some lectures. There must have been at least
100, maybe more. I mean, it's a usual occurrence.
I mean, they weren't just being polite. They were
they actually came up to me to say.
Okay. Anyway, 1980 was The Living Cell. It was published, and there was a debate with John Douglas of Brunel and Robards of York University, and somebody called Roberts of the John Innes.
No, John Douglas was in the chair. Robards and Roberts were the two.
Oh, Douglas on the chair. Okay. And Sartori was there, but ill.
Yes, he was
very ill, in fact.
Yeah.
Was that lung cancer, presumably?
No, it wasn't lung cancer. He had bronchitis and emphysema, so his lungs were actually destroyed.
Was he a pink puffer or a blue bloater?
He was a pink puffer.
A pink puffer. Poor chap. I saw a picture of him, in fact, in one of your quickets. He seemed quite nice. I mean, as far as we could tell from a picture, he seemed quite a nice chap. Okay, and these people, the tactics used against were multiple, including labelling your ideas as old hat, and saying that when electron microscopes were first introduced, all these things were raised then, and that wasn't true, or at least they couldn't produce any references to support it.
Yes.
And they started going on about things like, what do you mean by truth? And in fact, that must have happened before, because, as you say...
We wrote the book as a consequence, yes.
After the debate?
No, we'd written the book before then.
Yeah, but I mean, the book includes a lot of things about what is truth and Occam's razor.
Yes,
and that was
the reason was, you see, when we produced this book, this paper, which we sent to Nature, the Journal of Theoretical Biology, Science, all the senior journals.
Yes.
And they wouldn't publish it, and they said silly things like, what do you mean by truth? What do you mean by artifact? And one or two kind people, who I don't know whether they're just being kind or naive as well, said to me, well, if you discuss what you mean by truth and artifact and how much artifact is tolerable, then that will strengthen your case to get it, and why didn't you write up as a book?
So you sort of gullibly put that in.
Yes, and then when we did, we couldn't get the book published.
Yeah, and everybody presumably said that, I remember you telling me this, people said that in science books nobody says it anyway.
That's right.
Yeah, so they kind of marked it out as being an oddity.
Yeah.
Okay, now, 1980, Living Cell was published. You say here that your policy in the 1970s has been to refuse to speak to school or undergraduate students. In other words, in all your lectures were at universities, presumably,
yes.
All learning societies, so-called,
yes.
because that might discourage them from learning biology at all. You changed your mind, presumably, through sort of desperation.
Well, I changed my mind because I found that 12-year-old children were learning about the endoplasmic reticulum.
Right. So that was, yes, I see. And that was quite a fast input, isn't it, actually? Well, the Nobel Prize was only 4 years earlier. Although, admittedly, when was it supposedly discovered?
What, the endoplasmic reticulum? About 1945.
Oh, right. Okay, I suppose that it reflects the rate at which people who write syllabuses get promoted or something.
But as a matter of fact, it's interesting that when they originally described the endoplasmic reticulum, they couldn't get it published either. They got it published in a medical journal.
That's extraordinary. Who were the discoverers? How do you remember?
Porter, I think, was one of them. Claude and Fulham, 1945.
Okay, so that was the first paper. That wasn't a prize or anything. Oh, right. So that must reflect.
There was a considerable doubt about it.
So it took at least 35 years to get into the official syllabus, or say 30 years or something like that.
Well, yes.
Maybe earlier, I don't know. Anyway, never mind. School Science Review, full teachers. Mr. A. Bishop, the editor, invited you to submit a manuscript, and you submitted 11 pages, a re-examination of the fine structure of the living cell and its implications for biological education, which I presume was essentially the same thing, plus comments about its implications for biological education.
Well, what I did was, and again, one always gets attacked for everything. This man invited me to write it up, and I said to him, look, I don't want to write up the same thing because I've just got it published in Perception. I said, if you like, I'll write up the implications for science education. But in doing that, I obviously had to describe what main things I had said.
Yes.
And then I was attacked on the grounds that I'd said them too briefly.
Yes. You mean The Living Cell, don't you? That was the book you just published.
Well, it was both The Living Cell and our paper in Perception.
Right. Okay. Okay. It says there are two groups of electron microscopists: Horn, Harris, and then Michel, Finian, and Coleman took issue with us in writing. They acknowledged the help of a huge list of people: Professor Goslet, Robards, Burgess, Dr. Hunt, Professor Woolhouse, and so on. You got a letter of 5 pages to reply, and you just decided to list the 11 questions you'd raised. I believe in their reply, much of that was just to do with viruses and was somewhat irrelevant.
That's right, absolutely. Most of it was irrelevant.
Right, and only Michel replied to you after this.
I wrote to all not just those 5 authors, but all the people they'd acknowledged as well. And I said, I'd like to come to discuss these issues with you, and only Michel had the courtesy to answer.
And he said that there'd like to be more heat than light.
Yes. In other words, he refused to discuss it.
Right, he became an FRS. Okay, and what were the implications for biological education? Just to get back to the paper.
The implications were that one should look at living cells, that logic should be taught in courses, that students should be encouraged to ask questions.
The point about living cells is that you maintain that many students have never actually seen many biology students.
That's correct. You can get a PhD now without seeing a living cell.
Just having looked at micrographs and other people's work.
Yes.
That does seem extraordinary, doesn't it?
It is.
It's like being a doctor without having seen a body or something.
Do you know the Royal Microscopical Society recently launched a campaign to get a microscope in every biology class? Now, when I was a student, every student had a microscope.
That's interesting. I happen to know that I went to an Open University thing in June or so, and they had I sort of looked through a window and there was a biology class, and they all had microscopes. But I suppose that's a university, isn't it? Sort of school, I suppose that's like. Yeah. Anyway, so meanwhile you wrote an article on boxing. You were still doing this other sort of thing. Now, in 1981, BBC TV did their Horizon programme. No one will listen to me, which I heard here. You videoed it, which was rather insulting, really. It had a kind of voiceover. It dealt with. I think you were kind of overemphasising your part in it. Really, there were at least 6 or 8 people, including.
No, there were 3.
Well, there was there was a dead chap, wasn't there? Beringer, the fraud. There was something about Feynman. In America, there's something about Robert Millikan.
Oh yes.
There was something about a chap who was a fan of Uri Geller.
Yes, no, of the 3 contemporary cases they took out.
Well, yeah, but out of the total.
That's right. Yes.
Out of the total.
It wasn't that much, but I must say they did really do a very dirty trick on me, as I told you.
Yes, well, from my point of view, it's hard. I don't know what the contractual obligations were or anything.
There were no contractual obligations.
Did they get you to sign a release saying that they could use your interview in any way they wanted?
No, they just took it. And they're very careful not to do that because they didn't want to get into a legal situation.
Where did they interview? Was it the Natural History Museum, wasn't it?
Oh well, yes, and the Natural History Museum.
So they just turned up one day?
No, they asked me to come there.
To the Natural History Museum?
Yeah. There was a meeting of the Quecket which meets there.
Oh, right.
And they said, when will you next be in London? I said, I'll be in London next Tuesday for the Quecket meeting.
And the other time was at at Guildford they came
Yes, but you know what they really did, which was a very dirty trick indeed. You know the Natural History Museum, there's a huge
to.
dinosaur.
Photography about dinosaurs, yes, I know, yes. You didn't think of that, did you?
No.
You don't think of these things.
Yeah.
And they came to, what was that still at that stage? Oh, it was a different Unity lab, wasn't it?
It was a different room, but it was the same.
And they just turned up with their cameras and so on.
Yeah, they told me they were coming.
And you had, how long did they interview you for?
About an hour, I think.
An hour. So you ended up with about 2 minutes or something. I'd estimate.
No, about 10 minutes altogether.
In the programme?
Yes, but there were between
Nonsense. Nonsense. Your appearances were, I should say 2 minutes is an optimistic estimate. You said, yeah, you're overestimating your
No, but
I
mean, no, you're talking about my appearance, but they talked quite a lot about my ideas before I appeared.
Well, it wasn't a third of the whole programme, sure.
No, it wasn't a third.
I should say.
Oh, no, how long was the programme, was it?
I think it was a 30-minute one, I'm not absolutely sure.
Yes, anyhow, it doesn't matter, it was about 3 minutes.
Believe me, you're overestimating your partners. But anyway, it wasn't a very flattering programme. I wonder why, did you get what was the genesis of it? What gave them the idea of making a programme like that for Horizon? Did electron microscopists go to the beam and say they should do this or something? Was it?
I honestly don't know.
You didn't work out.
No, I never found out
the genesis of it.
Yeah.
Okay. Anyway, soon after the broadcast, they took you off all undergraduate teaching without any reason being given. You were an elected member of the Senate. The Department of Human Biology was closed down a couple of years later. Although I was the second senior person in the department and had London University degrees in medicine, physiology, and biochemistry. I was the only. When did you do a degree in biochemistry?
My PhD is in biochemistry.
Oh, I see.
Yeah,
I was the only member of that department not placed elsewhere in the university. Was that a strategic move, do you think, to sort of?
Well, what I hadn't said there was that at that time I was the chairman of the union, and the vice chancellor wanted me out of the way for that reason. He just wanted to sideline me.
Anyway, so you're now aged 40, and you now embark on 5 years' work on the structure of the brain. Roughly speaking, about 1980 to 1985, I got down here. I put this together out of what you said.
Well, yes, that's not quite. I mean, I've been working on the structure of the brain all the time, taking out single nerve cells. But what we did is we did a systematic study of the human brain and spinal cord from post-mortems. I don't think anybody will ever be able to do this again, actually.
Why not?
Because of AIDS. You see, we did, it's very complicated. In order to handle human tissue, you have to have special laboratories now.
Is it really? Extraordinary. There was some money in it for somebody, no doubt. Right, 1983, you published Biochemical Cytology, it's an interesting point. Let's discuss this for a second, and we'll get back to the nerve cells and naked nuclei and so on. Biochemical cytology has it advanced in the last 35 years now. It's very difficult for a layman to judge. We must discuss the question of progress, right? Now, at the beginning, in my scene-setting attempt, I discussed the difficulties of assessing technology, particularly if it's backroom stuff. If it's familiar. I mean, nobody's puzzled about computers anymore.
No.
But of course they were when they were big puzzling things. Similarly it's hard to assess technology. It's also hard to assess progress in medical fields, and the impression you certainly get is that there are huge breakthroughs.
Well, that is exactly what I disagree with, and I published 2 papers in the practitioner.
I've got another listed on my list. The problem is that from viewing it skeptically as an outsider, you find, for example, in the case of Ivor Catt, he thinks that because he was sacked from his job as a computer designer, therefore the computer industry should collapse. I mean, that's my interpretation of what he says. Perhaps that's being a bit unfair. So people could say of you, well, here's a critic of the system. He's bound to say that, from his point of view, he's going to say nothing's advanced.
Well, that's a very good point. What happened was that I have said that since, firstly, the techniques they were using had not been controlled. That's my first book. You see, had not been controlled, and many of them had assumptions which were contrary to the laws of thermodynamics and physics. You see, that's the techniques. Secondly, I showed with the electron microscope most of the things they were studying were artifacts. Therefore, I put forward the hypothesis that this was why there had been so little advance in medical research. Now, people said, oh, there have been lots of advances in medical research.
Yes.
And I distinguish between the technological advances, things like blood transfusion, penicillin, intensive care units, which were technical advances.
Well, let me take two examples. Intensive care units and transplantation.
Yes.
Because you might, both of them on the face of it are not just technological, they require knowledge of immunology, for example.
Yes,
on the face of it.
Yes, but the basic immunology was known long ago.
That people reject tissues under some circumstances.
That's right. But in other words, what was working out, it was a technology like, for example, cardiac bypasses. The anatomy of the heart was well known. It's just
But surely, I get the impression reading Medawar, one of his popular books, dated about 65 or something, that immunology, there's a whole lot of stuff about antigens and things, and it makes it look as though it's fearfully modern and up to date.
No, the theory of antigens goes back to Ehrlich in 1860 or 1870.
So that's not...
No, and that's the interesting thing. What I'm saying, and I've listed them, things like schizophrenia, Alzheimer's disease, multiple sclerosis, cancer, the genesis of them, the origin, how they start, is not known. And there are more people alive doing research on this than there have ever been in history.
Right. So your articles, these two articles on lack of advancement, make the same point basically. You say that if you look at apparent advances, or real advances, and you look at the roots of them, and you say it's the same old technique, but it's an improvement of technique.
Yeah, it's an advancement of technology.
Yeah, it's like looking at a modern tractor and saying it's got an old internal combustion engine or something.
I mean, that's right. I mean, if you take a modern car and the first internal combustion engine, they're both the same principle. One has improved technology very much.
Okay. So that's the... All right. Were these convincing? What replies did you get to these papers?
I got a lot of invitations to give lectures. But nobody has addressed the question of the extreme lack of success of research in these areas.
Okay, the areas which you've mentioned, things like schizophrenia.
Yes.
Cancer.
Yes. Nobody has...
multiple sclerosis.
Most of them don't admit that there have been no success because they deliberately mix up what I call the technological advances and the theoretical advances. But you understand, if one knew, for example, the biochemical genesis of schizophrenia, you could design logical treatment for it.
Well, you could at least try, yes, I understand what you mean.
Whereas you can't design it unless you know that. And that's what everybody's looking for.
But they're looking the wrong way.
Yes.
What about the other 1/3 of research that's going on in these places? Like, for example, I don't know, there's a place in Cambridge, or the MRC labs or something.
Oh, there's lots of good work. I mean, especially epidemiology.
Well, you mean just statistical work?
Yes. Well, it's perfectly good research. There's research on cancer in which you take animals, you paint cancer materials on them. There's research on physiology.
You count that as empirical work, would you?
Precisely. There's a lot of work on physiology. The physiology of walking.
Yeah, you measure people's. That's what I asked you before. With breathing and stuff.
That's right.
Measuring calorie consumption.
That's right. That's perfectly empirical. Very good stuff.
How is it? Let me just repeat. I asked this question before, and almost the same terms. You have tests on, like lethal dose tests and testing pesticides or whatever, all this sort of stuff. So you're saying that's perfectly good work?
Most of it is, yes.
Well, suppose, do you have in your criticism of this other stuff, I mean, are these things separable?
Yes, I call it basic medical research. You see, basic medical research, in my understanding, is research into the mechanisms causing disease.
Fundamental research.
Yes.
That's another word for it, isn't it?
Yes, that's right.
I mean, basic is often used to mean...
Yes.
It used to mean simple or not very subtle stuff.
Well,
but you don't use in that sort of
No, I mean fundamental, that's right.
Okay. All right.
But if I may say, if you say to somebody who's a well-known research worker in cancer, or if you say to Woolpert, for example, he's not a well-known researcher, but I mean he's a biologist, if you said to him, why do you think medical research has been so unsuccessful? First of all, he would say it hasn't. It's been very successful.
Yeah.
And secondly, if you pinned him down, you say, well, why is it we don't know the genesis, for example, of cancer, when there are a huge number of people working? He would say it's very complicated.
Yes.
Now, I would say that that's not good enough, because if you're using rotten techniques and looking at artifacts, of course...
You think that's similar to the comment I made earlier about people saying AIDS is complicated, whereas they don't say TB is complicated.
No, I don't think it is
it's
quite.
solved.
I don't think it is quite.
If they found a solution for schizophrenia, if they found an elixir of anti-schizophrenia, purely empirically, then it would be cured, wouldn't it?
Well, that's exactly what's happened with manic depression. See, lithium works on manic depression, and it's very cheap, and it actually works. And there are, I used to do it, a lot of people doing research on how lithium acts.
And also on things like the dosology.
That's right, and the toxicology and that sort of thing. Fine. But I said in public, I said it to my research assistants, I've said that it's a very good substance, lithium, because if we could find out how it worked, we'd get the key to manic depression.
I presume that was discovered
by accident.
Just by accident,
Yes.
Okay, let's see, 80 to 85, 5 years work on the structure of the brain through by doing post-mortem work, which you maintain might never happen again, since you believe in AIDS, naively. Right, I've got here a couple more discoveries that you made. There are only nerve cells, this counts as a rediscovery, I think. Only nerve cells and naked nuclei in the ground substance in the brain and spinal cord.
Yes.
Rediscovery of von Virchow, is that right? Or something similar to that.
Yes.
Also, there are no anatomical synapses.
Which is a complete discovery. So
that's a discovery, right. Well, 1846, I've got a little note here, taken out of one of your books. The great German histologist Virchow gave the name, the construction of the name. And this is where you describe the general consensus, where you've got all these 4 types. Astrocytes. I mean, they're obviously they give them these silly names, don't they? Oligodendrocytes. It sounds very impressive. A few bits of a few twigs, oligodendrocytes,
that's right.
A few twig cells, microglia. Okay, the neuroglial material is believed to consist of the 3 latter types of cells with very little space in between them. Right. In other words, it's crammed full of little tiny processes of cells,
cells and processes.
Yeah, with a very intricate fine network which takes up most of the space. That's the believed view.
Yes.
Right. You conceived the idea that Virchow right now, you get to here, there's a whole lot of things which go together. There's this staining business. You had to do a whole lot of controlled experiments on staining, which you published quite a bit later.
Yes.
There's also, I did a series of experiments lasting another 5 years. They all supported the following conclusions. Okay, you also did your sort of Occam's razor approach of trying to actually identify these cells, and you discovered that most neurologists are not confident in identifying types of cell.
That's right.
They can't say what they are.
Yes.
And you supported this in your film, in fact, your colour film, by showing, well, one of the things is just a vocabulary analysis, and like they describe astrocytes as a whole lot of different words, which appear to be
overlap completely.
Well, overlap, or not so much overlap as disjunct,
Yes,
completely different kind of expressions. Okay. Right.
And in addition to that, you see, what I did in my book on the structure of the living brain, the big yellow one, I actually I got their descriptions, you see.
Yeah.
And I show them all to overlap. Every single kind of cell. And then I got another table.
That's the staining, you mean?
Yes, but then I got another lovely table showing the people who'd actually described them said they overlapped.
Right.
They said they couldn't distinguish between them.
And nobody seems surprising that nobody's noticed that, or
it's very surprising.
It's just a kind of habit.
That's right.
You have your row of bottles on the shelves, and you get
right.
used to the chap tells you that this is used for so and so.
That's right, exactly.
Like you might like a housewife might believe that gin is good for rheumatism or something.
Yes, absolutely. Well, I was not long after that came out, I gave a lecture in Copenhagen, and afterwards a quite well-known man called Christian Krohn, who's died now, is an expert on the blood-brain barrier, and he came up to me and he said, Harold, he said, very, very interesting. He said, if you're right, you're for the big one. So I said, what big one? He said, you're for the flight to Stockholm.
Yes. Everything's wrong, wasn't he? Well, so far, anyway. You see. Okay, you were also unwise enough to find the evidence for the existence of the synapses contain so many inconsistencies that they're likely to be staining artifacts. Okay, 1985, anatomical synapse by light and electron microscopy paper. Okay, so there are a whole lot of things which come together here. The point about synapses being that, there's a cultural thing about stains, isn't there? The Germans went for aniline dyes.
Yes.
And these, the Spanish contingent seems to have gone for silver and gold. I don't know if that's a result of their history.
Yeah, probably.
But, so you looked at these silver and gold stains, like people like Ramon E. Cajal and all
That's right.
and you concluded that they were depositions of silver.
Yeah,
and more,
I think everybody agrees they're depositions.
Oh, right. And you've got these cells or splodgy things with little, what look a bit like sperm sticking out of them.
Yeah.
And by looking at these things are meant to be synapses, but they don't have the.
Yeah, have a connection.
So there's all so it's all inconsistent. So you found that out. You also looked at the substance of the brain under an ordinary microscope. I mean, without doing too much in the way of staining, and found it looked different.
Yes.
So that's all that lot
That's
goes together.
That's why we wrote our atlas.
Yeah. So this went on. Let me see. Well, okay. I've got down here 1986 that was when you were 56. That you published the cellular structure of the mammalian nervous system and also the film, which didn't cover the whole. Well, I don't know how much of the ground it covered. The film was much shorter than the book, so to speak, if I can put it that way.
Of course, yes.
And it had two parts to it.
Yes.
The second part. Okay, the second part dealing with synapses, wasn't it?
That's right, yes.
And the first part was the structure of the brain, and what really could have gone along with it was the film about staining, which came out, another colour film.
Yes.
Which came out, which include time-lapse pictures showing the things actually kind of shrinking and so on, changing shape.
We also publish in papers as well.
Right. So now let's try and bring all this together. So that was 80 to 85, roughly speaking. We also looked at the transmission hypothesis. Now this was presumably related to synapses.
Yes. Yes, you see, the transmission hypothesis.
About the same time, the transmission hypothesis is doubtful. This was depended on the existence of synapses. Transmission hypothesis, it's a slightly misleading name. It sounds more general than it is. It's really only to do with the supposed transmission across a little tiny gap.
Of one nerve cell to another.
Yeah, the synaptic gap.
Yes.
And of course, if you've got synapses, then you can look at the transmission hypothesis. If the synapses don't exist, what he says here is, I analysed transmission whereby signals are believed to pass from one part of the nervous system to another, and concluded that the view that transmission was chemical, formulated in detail by Professor Sir Bernard Katz, 1969 was his inaugural lecture on that, I think, contained too many unproved and unprovable assumptions for the theory as a whole to be acceptable. And 1991, well, it was a long time in the future, from our point of view, re-examination of the vesicle hypothesis. That's one of his 4 papers published later on, looking at the whole concept of the transmission. Yeah. So far, no one has addressed my objections to the theory. I felt it a duty to propose a new hypothesis. That was also published in 1991, another one of these papers. Now, between 1980 and 85, various other things happened. There was Parrish replied. Parrish reviewed the living cell. Harold wrote simple micro-manipulation and microdissection. Yes, a Cricket Club Journal article, and gave a talk on that typical kind of thing. In this case, at the Cricket Club, about the lack of need for fancy equipment. Really, you make your own stuff. And vegetarianism. He wrote an article on vegetarianism. 1982. Some questions not answered by electron microscopists was published. Also, there were some books published around this time. Broad and Wade, Betrayers of Truth. I got down 1982. 1985. Right. Now, at the end of 1985, we have the first book of a new series, the first paper in a new series, The Anatomical Synapse in Mammals by Light and Electron Microscopy. And this was the next stage on these, after the cellular structure of the mammalian nervous system.
Yes. What I did in that, you see, I showed that the synapse, the anatomical synapse is an artifact. But what I didn't want to do is I didn't want to put it in the title, so I just said the anatomical synapse by light and electron microscopy. I didn't put in the title that I had concluded it was an artifact.
Right, in other words, the title is subtly misleading.
No, it's not misleading.
Well, I mean, it's an edited title, so to speak. Okay. Also about this time, you saw M.I.M. Noble about Huxley and Hansen's sliding filament hypothesis of muscle fibres.
Yeah.
And 10 years later, you decide to write a joint paper when Brian Freeman comes up with not identical, but
other criticisms.
Numbers of criticisms about this particular hypothesis. And anyway, so here we go. So 1986 was your next big book, The Cellular Structure of the Mammalian Nervous System, which you could view as being a rediscovery of von Virchow's theory. That's perhaps a bit unfair. And it includes, as we said, material on staining, both the colour stuff and the silvery stuff.
Yes.
And concludes, there's a whole lot of, the whole lot of stuff. The first chapter is called Landmarks in Cell Biology. It includes a huge section of things like mitochondria and Golgi bodies, which to the casual observer looks like a summary of what's discovered, but in fact is meant as a kind of debunking. And then there's methods of studying neural structure, which includes these various dubious techniques.
Well, what I've done is that I think it's absolutely central that people should understand the methods by which the things they believe were discovered. Because a lot of people seem somehow to think that what they believe in is independent of how it was found out.
Yes. In other words, well, it's not surprising, it's the way it's presented in books.
I know, but...
And if you want to pass your exam, it's a sensible attitude to it.
Absolutely, but I mean, for a learned book, people actually don't know. If you stop the average person in the street and say, how do you know that, average biologist, how do you know that the DNA is in the nuclei? The majority of them would say, who know about it, would say, oh, by subcellular fractionation. And you say to them, have you ever considered what happens during subcellular fractionation? They haven't.
Okay. The point there being that it's the density that matters or something, isn't it? In other words, if you do subcellular fractionation of anything, really...
Yes, right, it separates according to density. Right.
Yeah, so the things can migrate all over the place, like this sandy stuff. Go to the bottom, wherever it was in life. Right.
That's right. Yeah. And also, ions and things which activate and inhibit enzymes can move easily. So what you find out at the end doesn't reflect what it started at the beginning.
Right. Now, there are another 12 chapters with cellular structure, including the practical recommendations. There's some appendices, including one on specificity of staining procedures.
Mm-hmm.
It's too much to go through there. Briefly, it ends up with unsatisfactory terms, another one of these collections of evasive expressions and a few naive questions at the end.
Yeah.
Incidentally, the same year, Cohn's False Prophets was published.
Oh, yeah.
So was Intellectual Suppression, edited by, among others, Brian Martin.
Yeah.
There was also a book by Michael Gold called A Conspiracy of Cells on HELA, Henrietta Lacks cells.
Ah, yes, but that was for a different reason, because they got into all the tissue cultures all over the world.
That's right, a scandalous situation.
Yes.
And also 2 Germans got a Nobel Prize for so-called ionic channels.
That's right.
What do you want to say about your big book, apart from the fact it didn't sell very well? Cellular Structure. Do you have any regrets?
Well, first of all, it was heavily overpriced, because it was camera ready. Heavily overpriced, and there was virtually no attempt to get it reviewed. But the 2 people who did review, one was neutral, the other was very, very laudatory.
Who was the laudatory one?
He's a professor of biochemistry at Galway.
Galway, right. I mean, that's listed in somewhere in your writings, is it?
Probably.
I remember you saying, mentioning there were 2 reviews.
Yes, but I mean. He should have been. I mean, you understand it presents such a different view of the brain that it should have been reviewed in any of the brain journals, and it wasn't. Right.
At the end of it, you haven't got a model of. I know whenever at your talks, you always say that you don't know what, you're not willing to discuss consciousness, thinking, that kind of thing, or the soul, the spirit.
Yeah.
So presumably, your view of the brain is not. It's just the biology of it, so to speak. It doesn't attempt to explain what goes on in it.
No, absolutely.
If anything, I mean.
Yes, absolutely not. Yes.
Okay. So, do you have any more comments to say about it?
Well, I was very annoyed that it wasn't reviewed, because, I mean, if I'm right, an awful lot of people are wasting their time. And I've dealt with some very important questions. We did original experiments which nobody's ever done, to scan the whole brain to see how much of it stains, with each of the stains which are supposed to stain specific kinds of nerve cells.
You did the whole brain?
Yes.
You don't...
Different parts of the brain.
Yeah, right. And this appeared in your the colour film came out next year in 1987, Effects of Staining.
Yes, but that came out of the book, actually. It came out of the book on the malaria.
Who was the chap showing you this, actually? Was that David Jarman? There was a kind of thinning on top chap shown. That was Dave Jarman.
Yes, I think so.
I wondered because it didn't look quite like him. How did your film go down? Cellular structure of the human brain.
Oh, tremendous hostility wherever I showed it.
Did you manage to show it anywhere?
Oh, I showed it lots of places. You see, because of the Physiological Society, Biochemical Society, Society for Experimental Biology, the European Society of Neurochemistry. I was a member of all these, and they couldn't stop
Once they
it.
showed it, you had a sort of evening, and your name was down there, and it was written up and so on, and people came along. It was advertised months beforehand. Sort of.
Oh yes, well, you send in an abstract.
Yeah.
And
I was just trying to get the feel of what the actual event was like.
What it was like, for example, at the Physiological Society, I showed it. There was a classroom about this size. It was packed out; you couldn't get in. I showed it at the Cambridge Biochemical Society, which was packed out. There were about 200 people in the room, and I had to show it twice because there was a huge queue outside.
Right, and it was advertised beforehand in the usual timetable system.
Yeah.
Harold Hillman will new film. So.
No, it was just you give papers for 10 minutes, and it just had the structure made of brain film.
And you showed that in? Did you also show that in US and France?
Yes, I showed.
Really? You went around yourself with it?
Yeah.
In a film can.
Yes, I've got the films. I've got film and cassette.
You just imagine you turning up.
Oh no, sorry, that coloured one I've only got cassette.
All right.
The first one I've got films in.
So, and you got presumably people came up afterwards. What do people say to you afterwards?
Well, most of them were just very angry. I mean, that's what happened.
Well, how can you present such rubbish, sort of thing?
That's right. Absolute rubbish. He doesn't know that such thing. But when I went to Australia recently, the first day I arrived there, I was absolutely wrecked, but it was a long airline.
Right, this was in last year, 1995.
Yes.
October or something.
That's right, it was...
or November, something like that, wasn't it?
Yes, something like that, September. I can't remember. But when I got to Australia, it was a packed meeting.
You took that video with you, didn't you? In fact, I remember helping you make it.
Yeah.
With all complete works on it.
Yeah.
The complete filmed works.
But I mean, it was half of the people were simply tremendously hostile. You can't imagine actually what it feels like. Sometimes I wake up at night sweating, thinking what a damn fool I was for doing this, because everybody thought they were going to get the classical structure of the brain, the neurons and the neuroglia and all that sort of thing. And when I tell them that, there's only one kind of neuroglia, they get really quite upset. But I mean,
they didn't actually throw anything at you?
They were some were very rude.
Were you very rude back?
No, I was sometimes being a bit scientifically rude. But
you didn't call them kangaroos or something? I don't know if there's any...
No.
I see, how disappointing. Right. Anyway, never mind. 1987, so that presumably went on... Well, we're back in 1987, so now you're back aged 57. An advanced age, indeed. Colour film effects of staining, fraud versus carelessness, letters in nature. How to make medical research more successful. This was your other paper or letter on technology not being...
This was two articles in the Practitioner.
Right. And also the effect of hematoxylin and eosin, Palmgren's and osmic acid procedures on the dimensions and appearance of isolated rabbit medullary neurons in the journal for things with long titles.
Well, I'll tell you all about that. That's a very interesting thing. You see, everybody in the world who's a histologist knows that cells shrink when you stain them.
Yeah.
Everybody knows it. But they don't like you to tell them.
They don't like to admit the figures. Okay, 1988, your second receptors paper, Critique of Current Views on Receptors The New General Theory of Receptor Location and Action in Vivo.
Yes.
I've got a copy of all of certainly those 4 papers. Well, can you summarise that one? Because I couldn't make any sense.
Oh yes, receptors is the biggest industry nowadays in biology. Indeed, Sir James Black got the Nobel Prize for receptors.
Ah, you spoke to him, didn't you? He saw one of your papers when he wrote.
No, he wrote. No, he read. No, that was different. No, Sir James Black was recently.
Oh, sorry. Okay.
Horrobin showed him my book.
The one on the 72 one, on uncertainty.
No, the
Living Cell.
Which one did he show? That was before I got it published. It was Case for New Paradigms.
Oh right, okay. So he got his Nobel Prize recently.
Well, while he was actually reading it, the Nobel Prize was announced, and I didn't have the courage to ask him his comments.
I see. Okay, so that was James Black. What do you say about critique of current views on receptors and the new general theory?
Well, everybody in the world, except me, believes that drugs, transmitters, hormones, toxins act by receptors.
Okay, by receptors you mean in the synapse sense?
Yes, they believe that along the synapse are special kinds of substances called receptors which react. So when you have adrenaline,
yes, okay,
there's a specific adrenaline receptor.
You don't mean, sorry, do you just mean in the synaptic gap or along the length of the thing?
No, they're all over,
Right,
but they are concentrated. There are a lot more on the synaptic.
But they believe that they all related to nerves. I mean, they're to do with synapses.
No. You have receptors to all kinds of all cells. Have
okay, no, right. So they've generalised it to everything.
Yeah.
So receptors is now widened to not just receptors across the synaptic gap.
Oh yeah. Well, it was widened a long time ago.
Right. Okay.
Yeah.
So it's receptors. They could be anywhere. They could be in your tongue or.
Oh yes. I mean they
or something.
Yes.
Kidney, liver.
That's right.
All right. So what was your new general theory? How did you criticise?
My new general theory is that you see a very large number of these receptors have been isolated and sequenced. In other words, their structure's known.
When you say that, you're being tongue-in-cheek, are you?
No. They believe they have.
Yeah, but you don't believe they have. So you're being tongue-in-cheek.
No. Let me follow the argument. They have found them, and they're large. You see, what they regard as receptors are very large. They're an average of 3 times as wide as a cell membrane.
Presumably they'd have to be just to fit big molecules in, wouldn't they?
Yes, but they actually have got the molecules they believe them to be.
Right.
And they are 3 times the width of... So I've said, well, why can't we see them on the cell membrane, where you can see the cell membrane by electron microscopy?
Right.
And nobody has ever answered that question.
Okay.
And there's one only they've ever seen, that's the acetylcholine receptor. And I pointed out there are literally millions of substances which have got receptors. So you should see not just one lump of one size, which you do see there, acetylcholine receptor. You should see a Himalayan mountain range with all different...
Right. How do you know that millions of substances have got receptors? Because they have effects.
They say they have, I don't.
Right, you mean anything that has an effect is deemed to have a receptor?
Precisely. Okay. Not everything. A few things like alcohol and anesthetics.
Well, when you say they've sequenced it and so on, you're being tongue-in-cheek, aren't you?
No, they've sequenced what they believe to be the receptor.
Yeah, but you're being tongue-in-cheek, aren't you? Because you can't possibly believe that they've actually... how have they made this mistake of finding big molecules?
Well, I'll tell you how they've done. Because what they have done is they have got substances which they believe act on the receptors. You see, they mixed up. If you take a radioactive substance which you believe acts on the receptor. Example, they haven't done this, but it's a rather complicated, so I'll simplify it. If you believe adrenaline is a receptor, you get radioactive adrenaline. You would add it to the tissue, mash up the tissue, and extract the radioactive product, you see. And since this is a receptor, you believe it's a receptor, then the substance you got out, you then study its chemistry.
Right, so you do all this mashing up and you end up with a radioactive
concentrate, which they believe contains the receptor.
And then you analyse it and you try and work out,
yes.
And you find, if you're lucky, multiple copies of some chemical, and you say, right, this is a receptor.
Yes, exactly.
Okay.
Now, the reason why I believe it couldn't possibly be because everybody agrees that by transmission electron microscopy, you cannot see that receptor.
Right. Is that your only reason for disbelieving in them?
No. The second reason is that they would be lots of different heights.
Yes, right. So that's a variation of your scaling. One of your arguments against electron microscopy is that you compare it with light microscopy and sort of scale it up and down.
Yes.
And people aren't bothering to do this, and so
Yes.
all sorts of absurdities get through.
That's right.
This is a variation of the thing you're saying, that they're taking one little thing in isolation, and if they were serious and applied it to everything, it would be impossible.
That's right. There wouldn't be room for them all.
Yeah, right.
But nobody has ever addressed this question. This is why I put it in a list of questions. Why do we not see them on transmission electron microscopy?
So I take it your new general theory of receptor location just doesn't believe in receptors at all. It's something different from that.
It's much simpler than that.
So it's not a theory of receptor location at all. It's a new theory replacing receptors.
Yes, but it's much. But it's a slight extension of the word. If, for example, you took a piece of leather, and you took a piece of butter and a piece of a raisin.
Yes, I think we've been through this before, actually, with dough, actually. But,
yeah, it doesn't matter. If you took three totally different materials, and you reacted with each of the large range of chemicals on your
yes,
bench.
I know we're going to say they all react to some extent.
Yes.
Unfortunately, I remember you telling us before, and I still was unable to see what you were getting at.
Well, what I'm saying is that supposing we took a piece of dough, it will react to a different extent with a large number of different compounds. You wouldn't say they're different receptors.
So what?
I think that the living tissue is a very heterogeneous material, and lots of materials will react with it.
Yeah, so what?
So you don't need a receptor for each of them. That's the point. They think you need a receptor for each, if you like.
Right, so you think there may be some receptors?
No, I don't. I think there are no receptors. I think the material itself acts as a receptor, just as a piece of butter will act as a receptor to organic solvents.
Okay. All right. Well, let's leave it at that. Okay. 1988. The Vice Chancellor imposes voluntary, in quotes, early retirement, and the Unity Lab is, well, they try to close it. They don't actually close it because Horobin pays some money for about 4 years or something, I think.
Yes, that's correct.
But they succeeded not paying you any money.
Yes.
And that kind of drags on in an unpleasant way. But nevertheless, you fight back somewhat.
Yes.
And you get them to pay for putting in a new plumbing system, I think, for example.
Yes.
But anyway, right, 1990, your new book, Atlas of the Cellular Structure of the Human Nervous System.
91, wasn't it?
I think.
Yeah.
I think it's dated. I don't know. I don't know. Anyway, it's a joint product with you and Jarman.
Yes.
And I seem to remember that it was soon withdrawn from the catalogue by the publishers, apparently contrary to their contract or something like that. They were supposed to keep them in print for.
No, you were
5
getting
years
this
or
up.
something.
No, you were getting this up.
Right.
The Atlas is still in print and is still selling. I published at the same year.
It was withdrawn from their catalogue, is what I said, not that it had been withdrawn.
No, what happened was, I told you, was that there were other books which were published before then, which appeared in their catalogue, and ours didn't.
Yeah.
And when I questioned them about it, they said it was going to appear in some other catalogues, but I didn't follow it up. I don't know whether it did or not.
Okay. It was withdrawn from the catalogue anyway, prematurely. Isn't that a fair statement?
Well, I would have thought so, yes.
Yeah. What was in it? I've seen this book once, I think. It was a collection of photos.
Well, what it is, we've taken the whole of the human nervous system, except for the retina and the peripheral nerves, you see, because we couldn't get those off from post-mortems.
You're touching your elbow there. That's a peripheral nerve, for example.
Yes,
Okay.
that's the ulnar nerve.
Yeah.
And we took the whole, and what we did is because we've shown that histology, staining, causes such shrinkage and artifacts, we took out the cells without any staining and looked at them by phase contrast microscopy, which enables you to look at them in the nearest to life you can possibly get them.
Nice. Is this somewhat similar to what Vesalius did? If Vesalius had a microscope, he could have done this, couldn't he?
Yes,
because
anybody.
he had a picture of the nervous system, the brain
Yes.
with.
That's right. Well, that was macro, you see.
Yeah, but so it's not very different from that.
No, except that what we did is
You
we
have
teased them
photographs
by
too.
the technique of Houdin, you see, which nobody thought of doing.
What did you conclude? I mean, was the object to conclude anything, or was it just to produce photos showing things?
Well, it's a bit crafty, actually. For one thing, we put in the nucleolar membrane, lots of pictures of it.
Yes, I'm sure. Did you label it as such?
Oh, yes, of course.
Right. And did you have the nucleolonema in it?
Yes. Well, we
Right.
didn't describe the nucleolar membrane. Somebody else had described it.
Someone else had. How could they describe that without acknowledging nucleolysis?
Oh, everybody agrees there's nucleolus. What they don't agree is the nucleolar membrane.
How can there be a nucleolus without a membrane? Am I missing something here?
No, I agree with you. That's the point I made. but the fact is,
they think there's a round thing floating around in the nucleus without a boundary to it.
Precisely.
Like the great red spot in Jupiter or something.
Yeah, yes.
God, how bizarre. And inside it, it's got...
The nucleolonema, yes. Which
it's supposed to have funny things that change shapes.
Yeah, that was our finding.
That it changes shapes. That's not just an artifact of light, the thing being tiny and it not being able to make it out properly.
No, because I've got two photographs, only two actually.
You don't think that's the DNA do, by any chance?
Which is the
wiggling around inside it with a nucleolonema?
I do.
You do? Oh, good.
I put forward a theory to Nature. I wrote a paper about 20 years ago and they wouldn't publish it.
Am I right in saying that part of the motive of this atlas was to smuggle in your ideas, in other words, presumably it includes your view of the brain and as well as
No, actually, some people have thought that. It's not quite like that. The fact is that I showed, with David Jarman, all the cells we could see. You see?
Right.
Now, we did not see astrocytes, oligodendrocytes, or microglia. And one or two of the people who read it have attacked us for not showing them. And I said we didn't show them because we didn't see them. Ever.
I see. So a reviewer might have said, who was not sympathetic, would have said that they haven't even shown examples of the astrocytes or whatever.
That's right.
Yes, I see. Yes, I see.
But we in fact showed every element we saw.
And that included the granular material made up largely of mitochondria or whatever it is.
Yes, we put the illustration of that, which nobody believes in either.
Right, an illustration without comment of what they contained in detail, apart from...
We don't know what they...
Yeah, right.
Well,
don't know. So
no, I put on one of the pictures. I put the granular material, a large number of mitochondria, or the granular material consists of a large number of mitochondria.
What, without anything else?
That's right. Yes.
I mean, it's just mitochondria.
No, there are the grains as well, but the mitochondria we identify.
Okay, you mean mitochondria make up a high proportion of it?
Yes.
What you're saying?
Yeah.
All right. And so that was that reviewed?
One German journal reviewed it.
What did they say?
Not very much. I mean, I couldn't read it properly. It was a very short review.
What did it say in the publisher's blurb in their catalogue? Did you ever read it?
Oh yes, I can show you it actually. Absolutely.
Yeah. All right.
Where have I got?
Okay, we have here a academic press. It's a A4 coloured sheet in full colour. At last, the human nervous system, beautifully presented in as near natural a state as can be presently hoped for. Atlas of the cellular structure of the human nervous system. Harold Hillman, David Jarman, University of Surrey, UK. Features hundreds of photographs, many in full colour, depicting the full spectrum of unfixed human nervous tissue. A truly unique achievement. Mm-hmm. On the back it's got ordering information and another advert for a rat brain and a human nervous system thing. And here we are. Photographs included the neuroglia, the white matter, droplet fibres. This is another discovery of Harold and Dave's, I suppose I should say. And the fine structure of the nucleolus. Teased tissue and hand-dissected cells. Right, there we go. What about these fibres with beads on them, droplet fibres? What do you conclude?
Well, we didn't conclude anything. I mean, we just described them. I don't know what they do.
Okay, now we've just had a tea break, so I'll try and take up where we left off. Okay, we've just done 1988, Vice Chancellor. 1990, your atlas was published. 1991, the case for new paradigms in cell biology and neurobiology. Okay, it's got quite a number of chapters. It's basically a kind of mixture of evidence with your own experiences. It's a kind of rather old book in that sense.
Yes,
the title is a bit misleading, as always, with a Hillman publication.
Well, the title was originally Letter to Students of Biology of the 21st Century, and the publishers didn't like that.
Yeah, well, anyway, it's a misleading title, because there's an awful lot in it of anecdotes about visiting people and them telling you that they had to go to lunch, that sort of thing.
No, but I mean, that is, in fact, based on Dingell's book, the idea of Dingell, which is half the science and half the narrative, if you like.
Late Dingell, okay. Right, there's various things. There are quite a lot of tables, including one with misleading terms, which has grown considerably. Also one about unproved or unprovable hypotheses,
Yes.
which in fact there are only about 15. I was quite surprised how few there were. There's also a table 9 here, possible origin of artifacts as well.
Yes.
Things like well, they're not all that many either. Lamella of myelin sheath, Golgi body, lysosomes, endoplasmic reticulum, cristae, cytoskeletons, synapses, synaptic thickenings, synaptic vesicles, cross bridges between muscle filaments, discs of retinal rods, thylakoid membranes, and nuclear pores. There's a total toll. Oh, there are some missing, aren't there? There's the microtrabeculae, for example.
Yes, but I mean, if you compare that with, I'm sure you've seen this actually. I mean the
1890 now, so they're very similar, right? Okay. Chapter 23 is does the structure of the living cell matter? I said here, short and disappointing from my point of view, Harold doesn't consider how much of biology has to be thrown away, or whether electron microscopes could simply be switched to something more useful.
Well, that's not my problem, really.
Can they be switched? You didn't discuss viruses, did you? You have an opinion that viruses can be studied usefully with electron microscopes.
No, only for classification.
Only for classification, right.
The reason they can't is because round the viruses you always get what they believe to be a wall round it, but it's the same thickness whatever the size of the virus, and that's impossible.
Another artifact, yeah.
Yes.
Is that what they call the protein coat?
Yes.
Does that mean you don't believe in a protein coat?
No. You see those two, those are the different...
Yes, I know that, yeah, I've seen that. Okay, now you've got here, does the structure matter? There are 7 comments. Firstly, we try and aim for scientific truth, ultimately. Secondly, a lot of these people are wasting their time. Thirdly, the ultrastructure, the attempt to correlate structure with function, they might well be wasting their time. Fourth reason, the employment of the major techniques, they're wasting their time using these particular techniques, perhaps.
Yes.
Fifth point, well, obviously, better findings and so on. For example, here's your example of lithium salts and penicillin as well.
Yes.
And you can't expect planned accidents to happen all the time. Sixthly, there's a thing about cynicism and so on. Seventhly, there's a thing about funding as well.
Yes.
The argument seems to be that collectors are individually inspired by the belief that we're honest research workers.
Yes.
So we ought to be. So there are these 7 rather miscellaneous reasons.
Yes, right.
So how was this book received? It was published by Mellon Publications, which is a very minor press indeed.
That's right.
In fact, I found a similar book about Japanese anarchists, which was a PhD thesis, published in exactly the same way, with a sort of binding with the chap's name stamped on it in silver.
Yes.
And printed from a typewritten text.
That's right.
With no
That's how they
illustrations.
make a lot of money. They've got a huge... they've got 200 or 300 books on their this. As a matter of fact, the head of it, a man called Professor Richardson, who's a Canadian professor, was fired from his job because he's hardly ever there, because he's running this printing.
So they make a huge amount of money out of it.
I think so. He's got a publishing house in the United States, in Canada, and in one in Wales where they publish.
Are you sure they're not just accommodation addresses or something?
No, he's actually got people working there because I've spoken.
Really, right. Can I ask how many copies your book sold, actually? Are you allowed to say that sort of thing?
Which one?
Well, all of them, I suppose, really. How many did the living cell sell?
The living cell sold fairly badly, I don't know.
Right. What about, the first one we know about, what about the mammalian nervous system?
That sold very badly. It was overpriced, and in fact it's now out of print.
And what about the
Case for New Paradigms? They won't tell me how many it sold.
Okay. Well, anyway, so that was 1991. Also,
the Atlas of the set of Brain has gone quite well. I think it sold about 700, which is, for that sort of book is quite good.
700 for your Atlas, okay. So also, anyway, 1991, the new paradigm, the Case for New Paradigms was published. Also, your 2 other receptor papers You published Resistance to the Spread of Unpopular Academic Findings in 1991. Yeah. That was for a journal called Accountability and Research.
A new journal, yes.
And I think I'm right in saying this, in 1991, both of your 2 final out of 4 receptor papers were published, the re-examination of the vesicle hypothesis.
Yes.
And a new hypothesis for electrical transmission.
Yes.
And then in 1993, I've got a gap there. 1993, you started, around about 1993, you started, and you worked 3 years at the Schizophrenia Association in Bangor, 3 days a week.
Yes.
You examined about 150 patients, according to my
That's correct.
notes, which sounds like one a day on average. So.
It could have been, yes. We saw them, sometimes we saw 2 or 3 a day.
Okay, and that must have taken up quite a lot of your work
Oh, it did,
at the
yes.
time. And in 1994, you went, this was when Chan Pillay, the Singaporean woman who married an American
That's right.
and returned home supposedly in triumph, attacked you with great indignation.
She did, and so did this man from Charing Cross, Geary.
Geary. 1995,
Mm.
in Australia, Brian Freeman pointed out that cell resting potential may be an artifact of measurement.
Yes.
And I think I'm right in saying that your earlier books don't mention that, so that must be a new thing.
That was a new thing and a fascinating thing, because the implications of that are extremely important.
But you said just now, talking to me, that Gilbert Ling takes that into account. He's already taken that into account.
Well, funnily enough, Gilbert Ling didn't really want to discuss this.
Is that because it's his own pet thing?
I would imagine so.
You know, it scuppers some of his work, does it?
I
Well, I thought just now you told me that he managed to allow for it, or did he invent the business of silver chloride and so on?
No, he invented the intracellular electrode, and it can be used for a lot of things. For example, it can be used for picking up excitability, but whether it can actually be used for the measurement of the voltage is the questioned issue which he had never discussed with me.
Oh, right, I see. There's a difference between the thing wobbling, which is
Yes, which
is one
it does.
thing, and there's the static thing. Oh, right, okay. Fair enough. I've got to the end of my notes now, actually. You know, apart from a few things about the stakes are high.
Yes.
If I'm right, a very large proportion of experiments will have to be completed. Let's kind of stop there, and let's just do a reprise. We do that as a good idea.
Yes.
I've got section 5 here. We can go through, basically just through your discoveries one at a time. Perhaps you can just comment on them, react to them. Okay.
Mhm.
So we go back to your age, well, it's basically between age about 32 and about 65.
Yes.
Yes,
Long time.
that's right. Powers of 2, 32 and 64. There you are. Perhaps you'll live to
as a
128.
matter of fact, it's 40 years altogether, because I started research work in 46 and I'm ending it in 96.
Right.
No, not 46, 56 and I'm ending it in
What?
96.
Well, did you discover anything then?
But in 56?
Yeah.
I did actually, because I started resuscitating patients who had very low blood pressure. It would now be illegal to do, but I started.
Oh, right. And this was what grew into your sheep resuscitator thing, was it? Oh, right. Okay, so there's resuscitation, but let's get back to, 1962, published in 1964, terminal phosphate bonds of transducer, and also 1966 paper, the effect of visible light on ATP in solution.
Yes.
What do you have to say on these?
Well, I showed that the ATP, adenosine triphosphate, phosphocreatine, and phosphoarginine are sensitive to light, sound, electric current, and sodium, potassium concentration and centrifugation.
Right.
And what was interesting is that phosphocreatine and ATP, which are present in mammals, were sensitive at 37 degrees and not at 22 degrees, but phosphoarginine was sensitive at 22 degrees.
Okay.
And I did an enormous number of experiments on that, and I still think that's true.
When you say as a transducer, that doesn't mean, that you can convert it from anything like light into heat.
Well, I think that was the implication, because you see ATP releases energy when it hydrolyzes. So if light can make it release energy and sound can, that is a proper use of a transducer.
Okay. Well, normally a transducer, at least in the sense I use it, means that it's used for some useful property. I mean, obviously, in a sense, if you have a piece of black paper out in the sun, that's transducing because it warms up.
That's right.
But it's not transducing in the sense that you couldn't use it to make a sound with or something.
Well, what I was thinking of doing, I've got in my fun book, if ever I get around to doing, what I was thinking of doing was putting ATP in a cell and see if it generates more current, you see. And that could be a detector of light.
So anyway, what was the main effect, outcome, so on, of this work on ATP and so on?
The main effect was I made myself very unpopular I had greatest difficulty getting it published. In fact, the major part of that was the only thing I didn't get published. I got the effect of light on ATP, but there were 5 agents and 3 different substances, so there were 15 different variables, and I only got 1 out of those 15 published.
And that was never published?
No.
Yeah, I see.
But I sent it to an international exchange, which was the equivalent of publication, and it is in fact registered there. So although it wasn't formally published in a journal, it has been published.
Okay, now aged 37, you discovered the nucleolar membrane.
Yes.
Along with Sartre.
Well, strictly speaking, I didn't. I had a chap called Hussein who was doing a PhD with me, and although I actually saw it first and I showed it to him, I put him first on the paper because he was doing a PhD.
So what's the outcome of that?
The outcome of that is that there is a nucleolar membrane, which I've shown all over the world, which we illustrated in at least 5 publications, including our atlas, and which most people don't believe is there.
Did you find any other cells apart from neurons?
No. We found neurons and naked nuclei.
Oh, right. Presumably, did you look anywhere else apart from neurons, I should say?
We looked for everything we could see. You know, we took the whole brain.
Yep. Did you look at any other cells apart from brain cells, nerve cells?
Yes, we looked at brain, spinal cord, and peripheral nerve. That's the whole nerve system.
You didn't look at muscles, for example?
No.
Liver cells?
No.
So
Only the central nervous
so they might have nucleolar membranes in them?
They might do, but we never looked at them.
Nobody else has either, presumably.
Right.
Okay, now, a bit later on, published in 1972, business about important biochemical research techniques that have never been controlled.
Yes.
Right, and that was true then. Nobody would reply to it, and it's true today.
But it's been very influential in British biochemistry.
The book has?
Yes, people are very conscious of controlled experiments nowadays.
Are they?
Although they haven't done enough of them, they're very conscious, and they very often pay lip service without actually doing all the controlled experiments.
You attribute that to your influence, do you?
I do, actually. Yes.
Were you influenced by Popper, would you say? Or was it just, was everybody, was it not business?
No more than anybody else.
Okay. All right, now a bit later on, 1971, published 75, most of the structures and cells apparent by electron microscopy are artifacts.
Plus the Golgi apparatus, which was described in 1898.
Oh yes, okay. And that was because of silver staining, presumably.
Yes. Yes. This view has not been accepted by anybody else in public, although quite a few people tell me they have in private.
Has it influenced theory at all? I mean, do people place less... for example, there's a theory that proteins were made in ribosomes or something, which of course depends.
That's right. It hasn't affected theory or practice or belief at all.
So there's hundreds of papers still involved.
Yes, people still say that... in fact, I was teaching a student. I teach, I do a bit of cramming. And I had to tell this poor young lady that, proteins are synthesized in the ribosome.
So you were 45 when you said that. Must have been quite a major thing, I suppose. Oh, not 45, I mean, 45 it's published. Okay. Now, big leap, now in your 50s, mid-50s, when you looked into anatomical synapses as distinguished from synapses, and you went through Sherrington and so on, you discovered that they don't exist, and the transmission hypothesis, therefore, can hardly exist.
Well, I first of all found out, or showed, by evidence from the literature, that synapses don't exist. Now, synapses are nerve-nerve connection. It is to be distinguished from a neuromuscular junction, which I do believe exists.
Are there not also nerve-nerve junctions in the sense that a very long nerve, which, a single nerve...
But they're still synapses, where they connect their synapses.
They're called synapses?
Yes. In other words, if one nerve going next to another has no influence on it, as far as is known.
Yeah, what I mean is if you've got a very long nerve,
yes,
that's not just one cell as a rule, it can be a lot of several...
No, with a long one they have synapses at intervals. In other words, you have a long one.
I've got this problem of understanding, of trying to work out whether you all got your tongue in your sugar. Do you mean they've got synapses, they have a join? Or do you mean
they have a join which other people call synapses.
Do they have a join at all then?
No, they don't. They're next to each other.
One of them stops.
Well, it's partly a semantic problem. Supposing you take two wires and you join them around to each other. Yeah. Are they joined together or are they not?
Well, I don't know. You tell me.
Well, they both are or aren't.
Well,
I mean, in other words, if you take that plug there, there's just one wire twisted round another. Obviously, are in continuity from an electrical point of view.
But you're saying it's like a neuromuscular junction. There is definitely a junction.
Well, the nerve comes up to here, and the muscle is there.
There are two. Yes, but when you've got one long nerve, and you're saying it's...
When you have one long nerve, it's synapses. What they mean by that is that, supposing you have a nerve nerve cell in the brain, it has a long axon which goes down here into the spinal cord.
Just one.
Yes, and in the spinal cord...
Are you saying that long nerves are always just one long process?
No, very often they synapse on the way.
That means there's another nucleus further down with its own processes.
Another cell, that's right.
And that definitely exists in your view.
No
yes. Ambiguity about that.
No ambiguity.
And they look different under the electron microscope, presumably, assuming you could find where the join was. I mean, does anybody know where the join? It must be quite difficult to find one, presumably.
Well, it's very difficult to find them in the central nervous system. That's part of my case.
Right.
But what they do is, you see, I did mention this before, but Sherrington said there must be a connection because when we stimulate there, we get a reaction there. And then the light microscopist said, well, we can see some end feet, so that's where it must be.
Yes, he was using a sort of telephone wire model.
Yes, yes.
But you're saying, okay, I was just quoting this particular point. That I accept there are neuromuscular junctions, although I don't know what the junctions are. And I accept also that that if you've got a very long nerve, it can be split into several nerves.
Yes.
Which unquestionably connect up in some
way. Like relay, yes. But,
and that's what you agree with that?
Yes, but
just tongue-in-cheek.
But whereas everybody else in the world believes that nerve A liberates a transmitter which goes to nerve B, which stimulates it.
Right.
I believe that nerve A has an electric field around it.
Right.
And that electric field is conducted in the fine granular material in the brain to the next nerve.
And are long nerves? Do long nerves have fine granular material too?
No, long nerves conduct directly. They're like wires.
Do they have...
That's called conduction.
Yeah, what about between where they join up to another?
Well, when you say they join up, they don't join up. They're near each other.
All right, well, what happens there, for Christ's sake?
I just told you, electric current...
then you change the brain, okay. What in a long nerve, it's conducted across the gap, is it, without using...
That's right.
It's like
a broadcast.
Okay. All right. So that's so much of the synapses.
Yeah.
And you came across that as a result of considering silver-stained micrographs and just looking at them and saying these look peculiar.
I give about 6 reasons why there are.
Yeah, but I mean that was where it fundamentally started. Yes. You did research. Yes. You searched the not so much the literature, the
slides and
the iconography.
Yes. Yes.
Okay, and as a result of that, you concluded that the transmission hypothesis must be peculiar and doubtful.
Must be wrong because the synapse is a central part of it.
Right. So at the same time, the sliding filament hypothesis, you examined this widely reproduced picture of the sliding filament thing under electron microscopy.
Yes.
And along with other people, decided that must be wrong, and that's due for publication soon.
Well, it's not due for publication. I've started looking at it recently. I haven't actually finished all the publications.
Okay, there's a lot of...
But I'm doing it now.
Okay, and a lot of diagrams and things, no doubt. Yeah. Okay, anyway, then... Okay, then...
I'm doing that with Brian Freeman in Australia.
Now, this is all tied up with the effects of staining. As I presume, I don't know when you started this, to what extent it became formalised, so to speak, but in 1987 your film was produced about the effects of staining, and of course that was included in the mammalian nervous system.
Yes.
So what do you have to say about that?
Well, we studied the 3 of the best-known methods of staining, and they all showed considerable shrinkage and distortion. So we have said to other people, now you must look, whenever you use staining, at what it does to the shape and size of the things you're staining. But, you see, histology is used for 2 totally different purposes. One of them is to find out illness. In other words, you've got an ill person,
right,
and you've got a lesion, and you know empirically that illness is associated with that lesion.
When you say what do you mean by lesion?
A lesion is an ill piece of tissue.
Okay, so it's exactly analogous to the use of electron microscopes for viruses. You end up with something which may...
That's right. Empirically has been related.
Yeah, right.
But
you see something there which every time somebody with a disease presents
precisely.
You hope it
does. If you want to ask the question, what does a cell look like in life,
right,
you cannot use any dehydrating technique because they are mostly water.
Could you just briefly... Okay, you did a series of controlled experiments with sizes of the brain and you showed they didn't overlap. In other words, you end up with sort of 80% of the brain unstained.
Yes.
What's the significance of that?
Well, most people believe, and this was said in a in a special issue of Scientific American in 1979 on the brain. Most people believe that the brain is composed of these four elements. So if you stained them if you could stain them one by one, the whole area would be stained.
Right. But if, in your view, staining is okay, so one of your arguments is that if the thing ends up, 80% empty, then their model can't be correct.
That's correct. Things, yeah. And we actually showed that because we stained with the different kinds of staining.
But nevertheless, if you're right and that staining is not that staining is not specific, then that model of the brain could be correct.
No. It's just that it makes it worse because if it's not specific.
Yeah. The question I made there was that if staining is not specific, then presumably you could still have the traditional model of the brain, and some of this, if the stain, I'm assuming the stains are not 100%, don't stain them 100%, therefore it would still be possible for the traditional model of the brain to be true, even if the stains are not specific.
No, because if they're not specific, then a smaller percentage, when you add them all up together, a smaller percentage than we got would be stained.
Okay, when you say specific,
well, you see, they believe they're specific for the four different kinds of cells.
They believe that every single cell of the type ends up dyed, or just a proportion of them.
Yes.
That's what specific means. It means that
yes.
all of them will be dyed.
It means that each different kind of cell is stained by a particular.
Yeah, but 100% of them are.
If they were right,
yeah.
then they actually say this, that the expression he uses, the whole brain will be black. Black means stained, in other words. The whole brain will be black if we were to stain them up.
Okay, so your staining experiments have several functions. One of them is to highlight distortions and so on. One of them is
there were different experiments.
Yeah, and one of them is brain specific. It's to do with
no, the ones on staining, we did a very interesting thing, a very simple thing. We take a single nerve cell and put it under the microscope.
Yeah.
Photograph it. Then we add each of the reagents. That's what our film is. I think you saw the film, didn't
yes.
you? And as you add it, you see that particular cell staining.
Right. And then you keep the
same cell.
And you draw the size of it
that's
before.
Right.
Yes, that's right. Okay, and these experiments have not been done before. That's what you're saying. At least not as maybe in parts, perhaps.
Nobody's done these specific experiments at all.
Isn't it rather remarkable that nobody has done it?
Yes, very remarkable. Or since. Don't forget, my book came out in 1986.
Presumably, part of the reason for this is that the high status it's got by inventing a stain of your own with its own wonderful properties, rather than doing detailed work. That's why they've all got names for them, isn't it?
No, I think it wasn't quite that. I think that the people who actually designed them believed them to be to show up particular kinds of cells, but they did not claim them to be specific. They didn't claim them to show other cells. This is what I showed in my book, that what they think of as a stain for astrocytes, when it was originally described, they said it stained astrocytes, but it sometimes stained neurons. And the stains for neurons say they stain neurons and sometimes stain astrocytes. But since then, people who haven't read the original papers actually have believed that they alleged they were specific.
What I'm saying is I'm distinguishing between people who do that type of experiment and people who might do controlled experiments. Presumably there's more status in having a stain named after you.
Yes, but in those days nobody did controlled experiments. If Ramon y Cajal said that this stained astrocytes, everybody said it. They didn't ask him, well, does it stain neurons or microglia?
Well,
because he was too great a man. He said so. So it's in all the books.
Oh, you think it's just a authoritarian thing?
Yes, yes, it is.
What would have happened if anybody had said, maybe it doesn't, and tested it? Or is it impossible to know what would have happened?
Well, it is impossible, except that in my book, which you may have seen, you probably couldn't read them because they were too small print, but there was I think a 3 or 5 page summary of all the publications showing that one person would say that something is specific for astrocytes, another person would use the same stain and say it was specific for oligodendrocytes.
Yeah, I'm not sure the point you're making there.
Well, the point I'm making
complete confusion.
Complete confusion. I mean, what one authority thinks is specific to one kind of cell, another authority thinks it doesn't.
So his word would go in his lab, presumably.
That's right, exactly.
So you're saying that things now are similar to that, only
yes,
not so bad? Or...
Absolutely similar.
Okay, now let's look at this other thing. 1995, Brian Freeman points out to you that the resting potential maybe or probably is an artifact of measurement.
Well, no, there is a resting potential. One can't tell what it is.
It may be an artifact of measurement.
No, it's not a total artifact. The value might not be correct. It's not a total artifact.
Isn't it? How do you know?
I
mean, I'm just saying it might be an artifact of measurement. There might not be one at all, presumably.
There might not be one. I think there's likely to be one.
What, just because there are ions on one side and the
yes,
other? There's just a difference between them?
That's right, exactly.
Okay. What does that...
Well, this has important, because what he pointed out was, you see, what you do is you put an ion-containing electrode on the outside of the cell, and when you go into the cell, it changes its voltage.
Yeah.
And anybody would think, well, that shows as a voltage when you cross the cell membrane.
Yeah.
But the electrode behaves quite differently in the two different solutions.
Mm. Well, I mean, I'm really talking about the theory. So what difference does it make if there's no resting potential, or if the resting potential is different?
Well, it means that you can't measure any voltage across the cell membrane.
Well, what are the effects of that?
Well, the whole of physiology and biophysics is based on measurements of transmembrane potentials.
So what effects does it have if the potentials are deemed different to what they were thought to be before?
Well, it means that for example, the theory upon which Hodgkin actually got the Nobel Prize, the ionic basis of electroactivity in the brain, collapses.
Right. Why is that, and what other can you generalise why it's important rather than just picking out one example?
Well, because the whole of biophysics of the nervous system is based on these measurements.
So what effects does that have on anything?
Well, firstly, it means all the teaching is wrong, but it means all the values are wrong and all the calculations are wrong.
Yeah. So what effects does that have?
Well, it means that I would think that 30 or 40% of all biophysics is wrong.
So what are the effects of that?
Well, the minimum you can say is that people are wasting their time.
What uses are made of biophysical results at the moment, if any?
Oh, calculation of the theory of muscle contraction, of nerve conduction, of transmission, of viscosity, elasticity.
Okay, you're saying biophysics is used normally as a kind of backup, a bit like maths in civil engineering or something.
Right, that's right,
it's
exactly.
a kind of collection of techniques which
that's right, yes.
Try and apply. So
yes, it's the application of physics to biological systems.
So what's conclusions, if these figures are wrong, what sort of effects does that have? Does it
devastating.
Yes, like what? On what?
Well, as I say, the theory, everybody in the world believes they know how conduction occurs, how transmission occurs, how muscle contracts.
Right. So, okay, now your claim is not just that the things could be wrong. What are you claiming? Let me think. But it's actually impossible to get it right. You're not just saying that...
Yes, it may be uncertain.
All right. Now, there's only one other thing I've got here, that which I've classified as a rediscovery, possibly unfairly, published in 1986, that there are only nerve cells and naked nuclei in a ground substance in the brain and spinal cord. What do you have to say about that?
Well, when Virchow said that there were only nerve cells and naked nuclei, he believed it and everybody believed it. Since then, people believe they have advanced it by identifying the other three kinds of cells.
Right.
Now, whether I wouldn't call it a discovery, because we've shown that the other three kinds of cells don't exist there.
It's a quasi-discovery, exactly analogous to electron microscopy.
Yes.
Right. So you're back in 1850 or something?
1865. No, 1849.
I see. Okay, the other four discoveries are dated between 85 and 91, and these are your four receptor papers, starting with the synapse, then receptors, particular receptors, then looking again at the vesicle hypothesis of transmission, and then the new hypothesis for electrical transmission.
Yes, what you've left out there is our work on resuscitation.
Was that relevant to? Oh, I see. I'm sorry. Yes. Okay. We
I mean,
mentioned that right at the beginning.
Yeah.
You said resuscitation.
Yeah. Yes. Well, what we did, I mean, we devised a new method of resuscitation.
Okay. Right. What about these receptors papers? Are they now a complete series? Is that all you've got to say on them?
Yes.
That's your complete works on receptors?
Yes.
Right. And what's the reception been of them? What have you been through this?
Well, two of them, no. Yes, they were either published in Horrobin's journal, which is a medical hypothesis, which is a very good journal, but it's a theoretical journal, and a lot of people think badly of it precisely because it publishes papers like mine.
It's a fringe one.
Yes.
Yeah.
Now, Ling runs another journal called Physics, Biology and Nuclear Magnetic Resonance. It's a very good journal which he runs himself, and he puts his own papers in it. He put my paper in it, and he does something better than that, because after each paper he allows people who disagree to comment.
Right.
And in my paper he was so upset by it that he got 3 different people to comment.
This was the paper about resting potentials.
No, we haven't published that. No, this was a paper about various artifacts various artifacts in cells. But he did publish it nevertheless. But people say, there you are, you see, they're only in these sort of peripheral journals.
Yes.
They haven't been published in Nature.
Well, that's perfectly true, isn't it?
But I'm sure I gave you recently a paper which was published in Experientia.
Yes, you did.
Which pointed out there were no controls.
The Birkhauser or something.
Yes, Birkhauser.
Yes, well, it wasn't really papers, it was only one page.
It was a page, that's right, just one page. But what I'm saying is that I tried to get that into Nature, I tried to get it into Science, to force somebody to answer it.
And they wouldn't publish it?
No.
Well, I'm not surprised. I mean, I'm not tempted personally into the delusion that thinking that things like Science are good magazines. I think they're sort of rubbish. I mean, they'll publish stuff which is...
No, well, they're in the pockets of the establishment, or they are the establishment.
Yeah, but not just in the science sense, but in the overall social sense.
Yeah, well, I think they try very hard, but as I say, people, if you believe something, it's very difficult to approve the publication of something that shows that you've wasted the whole of your life.
Yes. But, yes. So what do you... Okay, I've got to the end of my list of things. I mean, okay, so I started kind of trying to do the scene setting.
Yes.
I went through kind of year by year up to your present age.
Yes.
65.
Well, I mean, I will summarize by saying that I have taken issue with so many different people that I can't possibly win. At the same time, I cannot get a proper audience for my publications, precisely because the respectable journals won't publish them. And although I've got them all published except some of the effects of the physical agents on the other high energy phosphates other than ATP, all the others have been published. They've been published in journals which are not prestigious.
Are they obtainable? And if somebody wanted to look your stuff up?
Oh yes, they're all obtainable.
Well, they may be in theory, but I mean, you say in any library, if somebody in Colorado wanted to get your books, would he be able to?
Oh yes.
Well, how?
Oh. Well, they've got a National Library Service.
I mean, he'd have to order them through some central system.
Well, I mean, there's a National Library Service here. You can get any.
Yes, I know. Well, in principle, it's just a nuisance. That's all.
Yes, it takes a little bit of time. But I mean, if they write to me, I have very strictly answered every single letter I get. I replied to a huge letter, which I got 3 pages long, and I replied with a 5-page long letter by a fellow who'd been teaching all his life, and he asked me some of these questions at the Queckett meeting last week. And I replied in detail, and I always do that, I reply in detail.
What about journals? I mean, presumably they must be even harder to get hold of than books.
No. You can get them from any science library nowadays.
But not the obscure ones.
You can.
You think so?
Yes, I mean, if you want the Queckett journal, you can get they can get you a copy of it. If you want any journal, they always get you. I mean, ordinary libraries will do it as well as science.
You've studied, or at least made some attempt to study the lives of kind of famous heretic types, for want of a better word, like Semmelweis. Have you managed to draw any conclusions, for example, about the timing? You know, it might be that once the old guard die off, maybe there's always a time lapse of 20 years or 50 years.
No, I mean, as I said, I've simply taken on too many enemies.
Yeah, I'm asking a different question. I'm asking, in your studies of, so far as they exist, of these people, have you come to any conclusions? Or maybe they're too diverse to be.
They are too diverse, but don't forget, most of them suffered very badly. I mean, Semmelweis was a typical example. He died in a lunatic asylum.
What is he a typical example? I mean,
well, I mean, Virchow, he went out of pathology into public health.
You mean Virchow was not approved of?
No, he wasn't.
Oh, I didn't know that.
He went into public health. Of course, Ehrlich and Koch, they did very well for themselves. They were rather unusual in that sense.
Are you saying they were great dissidents in their time?
No, they weren't dissidents.
Well, why is it unusual that they should do well for themselves?
Well, because the whole of the scientific and medical system has tremendous resistance.
So they were dissidents?
Well, in the sense that anybody who thinks of anything is a dissident because they're a minority of one at the beginning.
Okay, so I get the impression you've kind of dipped into these things. You haven't kind of got a general theory of dissidence, so to speak.
No, I don't think there is a general theory. I think each one is different. I mean, Pasteur achieved great fortune and fame. I think mostly he deserved it. Whereas, people like Mendel, I mean, he died unknown virtually. No Gregor Mendel.
Yes, his work had to be rediscovered.
Yes. But I mean, I'm not interested in fame. I'm absolutely certain I'm right, and the reason I'm certain I'm right is because people get angry when I discuss an ordinary scientific problem with them. And I don't know how much you've done of it, but any time you meet a biologist, he gets very angry with you if you told him the endoplasmic reticulum was an artifact.
Yes, I've had a bit of experience with this, just trying people in labs and meeting people around. For example, the one in the scanning lab at Guildford. I told this woman it might change her intellectual life chatting to you because you were about to leave, and she didn't turn up, of course.
No.
Similarly, I met a woman I was meeting. I think she was a teacher at an internet cafe in London. I told her something similar. She just, because she worked for Professor so and so. Whatever, and she just kind of said, "Oh well, goodbye."
well, you know that book, this atlas we had.
Yes.
There's a chap I know at the Institute of Neurology. He's a member of the Quecket. He's a technician there, you see, and he put up that leaflet I showed you on the notice board.
Yes. At
the Institute of Neurology, where there's a large pathology department, which is highly relevant to. And the head of the department took it down and he said, everything they say is rubbish. He didn't even want the book to be ordered by the library there. They told me in the library.
Where's the Institute of Neurology?
That's National Hospital for Nervous Diseases, Queen Square. It's the prime neurological hospital in the country.
I see. Yes, okay, fair enough. What do you think of the comments I made right at the... You obviously don't agree with them, the comments I made at the beginning of just presentation and
well, we've had a continuous disagreement about this presentation question.
Yeah, well, my point of view is different from yours, isn't it?
Yes, that's right.
I think, from my point of view, it seems there is some mileage in the argument that you don't seem to learn from experience.
We've discussed
this. It's hopeless trying to address.
No, we've discussed this thing before. I believe that if you say anything clearly, which people don't like, they get very angry about it. They won't consider the evidence, and however diplomatically you put it, if you put it sufficiently diplomatic that it's not obvious, then they will agree with some generalization. They'll agree, oh yes, there are artifacts in electron. Oh, I agree that there's some shrinkage. Oh, he's got some idea, some good points. But if you actually...
saying that might open the door?
No, I don't think it'll open the door.
Oh.
Because I think that...
So you're saying it might just as well be completely clear?
That's right. That's right. Because if you say it loudly and clearly, they get angry. And if you say it sufficiently mutedly, they can ignore it.
Right, so you're saying you should be loud and clear?
Yes, I think so. I've had arguments with my collaborators.
Well, I mean, I personally agree. I just don't think, I suppose perhaps we disagree on what is loud and clear.
But actually, I would love to set up another debate. I mean, there hasn't been a debate since 1980. I wonder if do you go to London Skeptics?
I went once. I could get in touch, I suppose. I'm not in touch particularly.
Yes. I mean, you see, at University College.
Do you think debates are worth going to? And if you've had several hundred in your life, it seems like.
No, I've only had two debates in the whole run.
Right. I suppose lectures, you mean?
Yes. Lectures are the same as a debate.
Okay, by debate you mean you have to get some opponent.
Yes. And they all run away. On several occasions, the BBC has wanted to set up programs, television people, and they've been, the particular example I think I give in my book is University College, where they told them that nobody believes in the endoplasmic reticulum, so that I was tilting against windmills. Can you imagine that?
Right. This was a Horizon or something like that.
Yes, one of these
science program.
Yes.
Supposedly science program.
Yes, it was the other one. What's it called? Not Horizon, but there's one like it called.
I don't know. Wouldn't be Tomorrow's World, would it?
No, not Tomorrow's World. No. It was like Horizon. Anyhow, not Panorama. Anyhow, but several occasions they've told the BBC that.
Okay. What about the problem, the very great problem that people are overawed by the huge mass of material? You know, there are all these hundreds of papers, hundreds of journals, all saying exactly the opposite to you.
Well, exactly. That is one of the reasons why you said to me earlier, well, what about the 30% or 40% of research rate, which is good?
In your opinion, might be.
In my good, yes. Might be. But even if they were all good, it's a very large noise-to-signal ratio. In other words, there's so much noise when there's crap they talk. I mean, Greenfield talks total crap, and I would never say it to her face. But if you could manage to get a debate with her, between her and me, I would frankly wipe the floor with her.
Really, it'd be interesting to.
But she wouldn't do it, because she's now part of the establishment, you see. She is a lecturer, I think, at Oxford. Is Oxford, Cambridge, not?
She's at Lincoln College.
Oxford.
I remember that from the letter you wrote. I think that's Oxford, yes, I'm not
yeah.
sure.
But, you know, she's at the establishment. You see, she writes articles in the Times, and the Sunday Times, and she...
Yes, you tell me what's that, you know, she's... Okay, last year, or early this year, she gave a talk on consciousness.
Yes.
And you said that was absolute crap. It's, you know, consciousness...
it has no physiological substrate.
Well, that doesn't mean it's crap, does it, though?
No, but she's a physiologist.
Yeah, you mean she's not entitled to say anything about it?
Yes, exactly.
Anything more than
I mean, it's like asking me to talk about transubstantiation. You know, I'm not an expert on it. And I certainly wouldn't appear on the BBC on it.
What about the problem, as I said just now, which you don't seem to have answered, that if you're a student or whatever, or a reader or anything, there's this huge mass of impressive-looking stuff with long words, you know, which appears to mean something.
Yes, well, what you regard a person like me as a bloody nuisance, because you ask awkward questions, might be true or might not be true, but you've got to teach students, you've got to pass examination, you've got to get grants.
That's right. Well, it's not just that; it's also researchers as well.
Yes.
But
well,
I mean, that's a serious problem. There's this amount of stuff, and it does look impressive.
Well, it's also the other point which I made to you several times, that if you spend 3 years doing a PhD.
You're not really addressing the question. Yes, I take your points about vested interest and so on. But you're in the. If you look at an establishment like the medical establishment, suppose you were, 50 years ago and you were combating the medical establishment about anything, fibre in food or something. You've got these huge volumes. They all say that you're talking rubbish, right? And they're all wrong. But the problem is attacking it and sort of self-confidence and so on. Do you have anything to say about that? I mean,
it's unfortunately it gets back to what I said before, that you can have one enemy, but if you've got 25 enemies, you can't win, however right or wrong you are.
That's not quite. If you look at things like contraception, for example, Mary Stopes had immense opposition.
Yes, but on one issue.
No, I see what you mean about the issues rather than people.
Yes, one issue. I'm quite sure that if I spent the rest of my life, I could show to everybody's satisfaction the endoplasmic reticulum artifact, people would laugh when you mentioned it.
from then on, you mean?
Yes.
Like phrenology or something.
Yes, or like, what's the name of that Frenchman who had special kind of...
Oh, Edenrays, there's the chap from Nancy.
Yes, Blondeau.
Blondelot, I think it was, wasn't it?
Yeah.
Yeah, but you're still not addressing the question that you've got this very impressive stuff, and somehow you've got to try and couple it, make it look ridiculous.
Yes, but I mean, I can only, let me tell you how you can do it. You do it at international conferences where they won't let me speak. You do it at lectures where they generally don't invite me, although I've given a lot.
No, you're not. Even if your lecture is successful, you'd still have this volume of material, and it would be impressive looking, and lots of students and so on would be faced by it, and
that's right.
people doing
But
research.
isn't it exactly analogous with a religious situation?
It's very closely similar because there's a money involved. In fact,
There's
we
a
discussed this
theology,
earlier.
yes,
No,
yes.
the theology has nothing to do with it. It's the money involvement.
Well,
That's not the
I
whole
wouldn't
point.
just put it in money, it's the power and the social organisation,
Well,
and the prestige.
That's right. So the theology, the kind of theory, has nothing to do with it, in my opinion.
Little to do with it, little.
Except in the sense, of course, it has to be disguised. You have fancy, you have elaborate words and phrases.
That's right, exactly.
If you look at, for example, the way Hegel was developed at the end of the 19th century, you get very florid sort of stuff.
That's right, absolutely.
And that looks impressive. But, okay, the question is, well, in the case of Hegel, in Britain, I don't know what happened in Germany, in Britain, Moore just attacked it, and everybody, Russell climbed down, and there was a general, it just kind of
disappeared.
But I mean, presumably that was partly, there just weren't that many academics around then.
Yes.
And the new ones came along,
yes,
something to do with the age groups.
But again, it's still a relatively one-issue situation.
Yeah, that's true. Well, it's sort of true, except that Hegelianism did claim to cover space, time, history, and all of human life.
Oh yes, that's true. That's true. But I mean, it's still one concept, really.
Yeah.
I
still don't know that I'm getting my point through, really. That you've got these papers, huge.
No, you're asking me how I could advance my belief.
No, I'm not. No, you're taking an egocentric point of view. What I'm saying is looking at it from a student, from everyone else's point of view. They look around. They're in a laboratory. There are huge bound volumes of papers. They anyone and books, numbers of books on learned discussions. Meetings on these synapse, expensive conferences that people go to, hundreds of them every day of the week, huge, great, massive things, full of, typewritten, photocopied, luxuriously printed things, all of these diagrams in. I'm not talking about you, I'm talking about them. From their point of view, how can they have to have some way through it. A sort of
well, the bottom line is intellectual honesty, isn't it?
But I don't know. I mean, it has to be presented. They have to have some way.
But look. This lady you met in the library, supposing she came to my lab and sat down, and I told her that all these things were artifacts, what would she do? She'd get very embarrassed. She wouldn't know what to do, because she's probably doing a PhD, and she's not going to give up her PhD on the endoplasmic.
So it's her limitations, really, you're saying that?
Yes.
You're not giving her the part of the problem is, though, that she doesn't have a complete... Well, maybe don't know.
No, but I mean, even if she did, supposing I go to her supervisor and I tell him, he'd get very angry.
No, but you're being sort of ego-centered again. You'll think about you doing things.
No, I don't. I'm not talking being ego-centered. I'm telling you the general proposition.
Right. Suppose.
All right. Let me give you an analogy. I've got a book over there which has got from two of the Gospels the lineage of Jesus from the house of David. And they're both different. Now you take that to a religious Christian and say to him, now look, this is impossible because you believe both books and they're both different. What will he do? He'll get very embarrassed and throw you out or want to throw you out.
Yeah, we're talking about a different thing, really.
No, you're not.
Well, imagine you're a student and you need to be, there are all these things which you're presented with implicitly in all these impressive looking publications, and they've got one or two of your books. What are they to make of it? How are they to respond? How are they to sort of deconstruct?
Well, I'll tell you, I had a very intelligent Danish man working with me, and he largely agreed with what I said, and he asked me, he was doing a PhD with me, he asked me what he should do in life, you see. And I said the best thing is for you to take up an area which, on mature consideration, especially if you agree with me, you've read my works, is not covered by this, preferably an empirical area.
Yeah.
You know, that's the only thing to do.
What did he do?
Well, he did, in fact, do that. He's a psychiatrist, actually.
Oh, I see.
So what he did a study of a large number of patients of different kinds. That's fine, empirical. You see, and you can look for the biochemical changes in that particular condition. You know, again, empirically, if there are any.
Yes.
And that's what I advised. Now, I had another chap who was doing a PhD with me, the one who we pretend found the nucleolar membrane. He was as dumb as anything. And I think I told you, he took out of my first book a large section attacking the electron microscope. And he was writing a thesis on isolated nerve cells, there's nothing to do with it. So I said, why did you put this in? He said, he thought it would please me. I said, but your thesis has nothing to do with electron microscopy. You haven't touched an electron microscope. So he took it out after I persuaded him to. And about a few weeks later, he came back to me and he said to me, could I get him a job? Did I know anybody who had a job in biology? I said, what? Did he want to go into it? He said, electron microscopy.
I see. That's strange.
And he's now a professor largely in electron microscopy.
Where?
In one of the Gulf states.
Okay.
But what I mean is that man, frankly, was so dim that it didn't really matter what he did in life. But the Danish chap was very, very intelligent, and I would have been very sorry had he gone into something which, you know, had he gone into subcellular fractionation.
Yes, I tell you, but I'm still I still don't know what to say with this. I'm give an analogy. If you suppose you're interested in history,
Yeah.
and you suppose you find out that in the 18th century there were continual, there was slave trading going on, and this huge industry of slave trading, and the traditional model of the 18th century is that there are all these gentlemanly people sitting around in salons and so on. And there's huge numbers of books on it, and history magazines and journals, and they all say the same sort of thing. It's just what do you do? What does the it's nothing to do with you. It's not. And somebody, you come across this in some way, analogous to reading one of your books.
Yes.
What are they supposed to do? I don't.
Well, no, I agree. It's a personal problem. But what you do, I think, is I can't answer for anybody in that situation because I feel very sorry for them.
It's not just you're quite missing the point, really. They might think they're wrong. They might start to think, well, I must be wrong about this, or
Well, that's what everybody thinks. They say, look, Hillman can't be the only soldier marching in time. That's, I think, quite properly.
So you don't.
They think there must be an answer to this endoplasmic reticulum problem.
So there's no solution to this general problem. You just have to try and read and think and all the rest of it.
Yes. And, I mean, a lot of people, including Ling, said to me, well, your enemies will die out. I think it's a famous Chinese saying or something, isn't it?
I doubt it. I mean, there's a whole industry of inventing imitation sayings.
That's right. But I mean, they say that, you never conquer with a new idea. What you do is you wait till your enemies die out. Well, I don't think that's true, because the more bad research goes on, the more people have a vested interest in the bad research. So I will never win, and I won't even win in several hundred years.
You think so? You think this will be going on in the 2300 or something?
I'm sure it will.
You think so?
Yes, I mean, for the same reason as all major religions are still going strong, aren't they?
Well, I wouldn't say they're going strong exactly. I think they have serious problems.
They have problems, but they're still very influential.
Are they?
Oh, yes.
I wouldn't go so far as to say that. They do get, of course, state aid and so on. So there are those analogies with.
That's true. But I mean, the Catholic Church, the Islam, they're
What do you
all...
think of Theocritus' idea? I mean, he favours privatisation, doesn't he? He thinks that research the government should offer. Do you remember what he said? I mean, his exact example was Harrison's chronometer. He said the government, they didn't offer money to Oxford and Cambridge to solve this problem of finding longitude. I think it was.
Yes.
Was it latitude, longitude? They said we'll offer a prize after the event.
Yeah, to anybody. Yes.
Yeah.
Well, of course, you have to be a man of leisure to be able to do that.
Well, it would be a sort of investment thing, wouldn't it? Do you have any views on that particular idea?
No, I don't like it, because it means it's like a competition for an architect. Or a whole lot of people put in a lot of work, and one person gets it.
That's right. So if you reject the Theocritus solution, what are you left with?
Well, you're left with going on clapping and feeling very sorry for the people who are wasting their time.
I confess I find that feeling highly misplaced. Hi.
This situation now.
So.
It's a believer situation, isn't it?
Yes. I mean, I don't. My model of religion is different from yours. You think that people actually believe what they say. And I think in religion, most people who are supposed believers, the belief is negligible.
But I think you'd agree that's the thing you can't establish. It's not a general answer.
Oh no, you can, because you can ask people.
Yes, but you see such a small sample. You see a non-statistical sample.
Well, I could easily do a statistical sample. You can easily ask. I've noticed it in any religion. If you ask C of E people what their views are, and you look up any aspect of the church, or the religion, or history of it, they don't know the first thing about it. Same with my mother-in-law was ex-mother-in-law was an enthusiastic Jew. And I asked her about things like the Aramaic-speaking Jews, and she doesn't know the first thing about it. And this is absolutely standard. The reason that they are so-called religious is because it's it's a kind of social thing.
I know, but isn't that exactly analogous with science? I mean, there's a whole religion.
Well, you're assuming that the belief system is relevant, but
no. A religion, we've discussed this before. Religion consists of about 5 different elements, but science does as well. There's what we can call the theology, the ethics, the organisation, the money, the people.
If by theology you mean mouthing a set of like a creed.
Well, that's exactly the same thing.
Same thing,
isn't it?
It's closely similar. It can't be quite the same because they do what looks like research.
It's not quite the same, but it's very closely.
Well, you don't get theologians doing what they call research, do they? Although they do write.
No, they do. They
write Sunday. Yeah. They write their parish magazines, don't they?
Well, no, they do research. I mean, you can do research on the ancient Israelites, and you look up all the references in the Bible. Then you look up any references you can find in Egyptian literature, or the tombs or the writing they have. What's it called?
Hieroglyphics.
Hieroglyphics, yes.
Right.
That's perfectly good research. It's, if you like, empirical research.
I suppose that is analogous to looking up back numbers of papers, isn't it?
Yes.
Although I can't think of any practical equivalent, although I suppose there are some. They don't have labs, do they?
Well, I mean, if you take a person like Karl Marx or Hegel, for that matter, I mean, they had a tremendous knowledge of history, and they made up theories of economics on the basis of their knowledge of history.
Bertie Russell said that Karl Marx, displayed great ignorance of history. But, I suppose it depends on what you regard as being a lot of historical knowledge.
Yes.
Oh dear, right. Well, you've got a few minutes left on this tape. Do you want to use them up?
I don't think so. I mean,
you've discovered. Have you noticed? Okay, in your discoveries, we've been through them. Mm-hmm. There was you started with ATP,
mm-hmm.
and then there's the general idea of controlled experiments not being done, and then electron microscopy. Well, it's part of the same thing, artifact. And then a whole lot of hypotheses to do with the brain.
Yes.
Is there any possibility you might discover a whole lot of other things? For example, to do with the way the body works, or something like that. Can you see?
Well,
10 years' time, you might see something.
I'm developing a theory on cancer, based on my findings in the brain.
Right. That's more. Okay, I remember you saying that cancer in the brain is able to propagate quickly because it's a syncytium.
That's
nothing
correct.
to stop it.
Yes. That's my theory. I mean, that's a.
Right. Nothing to stop it being. Cell membranes presumably being tough enough to have some effect normally.
Yes, that's right. Well, to resist. I mean, if you got a crowd, if you get off a crowded train, there's a crowd standing there. It's not easy to walk through.
Does that only apply to brain tumours, though?
No, that's what I'm developing a more general theory. Because I don't think there are lots of tissues which are syncytial, including muscle.
Oh, really? I didn't know that.
Most people don't consider them syncytial nowadays, but they used to.
The only other example I heard of was fungi, some sort of.
Some fungi are syncytial.
Right. So basically, you're going to look around at lots of syncytia.
Well, I've got a list somewhere of them, for example.
Right. And so you look at muscle sarcoma, that kind of thing.
Yes, precisely.
Right. So you might get someone with cancer, but then of course presumably you'd have the problem of of publishing it and so on.
Oh yes, yes.
Do you think you might find some tie-up there with supposedly crank views on... I mean, my brother's got the thing with the Hoxie Clinic, which is notorious in America for being persecuted, but it nevertheless seemed to work in some cases.
Yes. Well, I haven't heard of it, but I mean, one is continuously running the danger of being associated with crank, or being called a crank.
Yes, I know.
And the answer to that, again, is to keep your head. That is the biggest problem, to keep your head while all around you are losing theirs and blaming it on you, you know.
Good heavens, you've been reading Kipling.
Oh, yes, I'm a great admirer of Kipling.
You're a great admirer of him.
Oh, yeah.
Tariq Ali and Edward Said were discussing great literature.
Yes.
And they discussed Kim, and they seem to be mutual admirers of Kim. Can you believe it?
Yes.
I don't admire Kipling.
Well, no, Kipling wrote some beautiful poems. The fact he was an imperialist and a racist.
Well, name one beautiful poem he wrote.
I think If is a beautiful poem.
Do you? It's not exactly beautiful.
Look, I've got there a whole lot of poems. I'll show you one someday.
So anyway, cancer is a possibility. What about schizophrenia, multiple sclerosis,
No.
Alzheimer's disease?
No, cancer is the main one I'm working on at the moment.
Radiation sickness?
No.
Pesticides, pollution?
But, cell division I'm also working on. I've got a theory of cell division. It's too late to go into it, but I think it's questionable the current theory of cell division.
What with meiosis and mitosis and so on? Oh, right, with spindles and...
Yes, the spindle problem. I mentioned to you, the spindles are meant to pull the chromosomes to a pole, but since they're not attached to anything, they couldn't.
So there's some other mechanism?
Yes, and I'm trying to work out what that might be.
How are you doing that? By going through the literature?
Yes, first of all making empirical theory and then going through the literature.
Do you find literature searches...
Oh yes, very useful.
They're useful despite being disheartening.
But I don't do them on computers, I do them,
I mean, they must be a bit disheartening to see the same old stuff over and over again.
No, it is often, it is often.
And presumably there might be just one little clue tucked away that you could easily miss.
Oh, absolutely.
I mean, like the nucleolus, for example.
But let me tell you...
you could have discovered the nucleolus by going through the literature, couldn't you, in principle? But you just have to
no,
look
not
at
the nucleolar membrane.
Yeah, but you just have to...
Yes, but I mean, those were drawings, you see, we've actually got photographs of it.
Yes, I know, but you could nevertheless have...
Yes, I could have, but I mean, it's much more valuable to me to look down. And if you imagine, I showed it to people at the Physiological Society, and they said there's not a membrane there.
How do you know it was a membrane? You can just see the edge.
Because if you focus in and out, it's like a ball. It goes in and out of focus.
Right.
But do you know what somebody said to me? Somebody said to me, well, you haven't shown that it's composed of lipids and proteins.
Yes, I know, yes. So that's a diversionary move,
Exactly.
isn't it?
Exactly. But I mean, it shows you how much, frankly, they dislike me. There was another occasion where my assistant produced some, I think it was quite beautiful work. She did a PhD, and she gave a paper to the Physiological Society, and they rejected it because I was a co-author. There were 25 people present, and they rejected by 5 votes to 4.
The others abstained.
Yes, they usually abstain.
Why?
So they feel it's not their specialisation, so they don't express it.
So it's the easy way out as well, isn't it?
Well, yes, but I mean, it's also true that, for example, when people talk about kidneys, I know very little bit about kidneys, or genetics. So if it came to a vote, I would usually vote for the publication, because I don't know anything about it. If it was a bit fishy to me, I'd abstain. But it's not the central area of my knowledge, so I don't know whether it's right or not.
Do you know anybody else apart from Ling who's following the same paths as you are?
There's a man called Schwaber. Now, Schwaber is a very interesting man. He's shown that Darwin was wrong.
Oh, this is a chap in America?
Yes.
Oh, with the 2 different evolutionary trees.
That's right. The evolutionary trees based on the biochemistry and on the zoology are different. And that's impossible.
So he's wrong, is he?
No, he's right.
So you're using these strange expressions again?
He's right.
Is he? Yes, that's one of
His name is Christian Schwaber.
It's one of your stock examples, isn't it? Like this
yes.
chap in Florence.
Yes.
You have discouragingly few stock examples, unfortunately.
Well, I wouldn't say that. And there are very few people who are prepared to stick their necks out. I mean, this fellow, although he's a professor at the University of South Carolina, he'll never get anywhere. Well,
Never in the whole of his life.
Yeah.
You've seen it happen before.
no, I have. There's a very nice man called Good who has a theory of muscle contraction, you see, and he absolutely slates the sliding filament hypothesis of Huxley and Hansen, you know, the current view on muscle. And he produced a beautiful book, and this book is based on the belief that the electron microscopy is correct, and it isn't. And I had a nice correspondence with him, but I didn't want to break his heart by telling him that I don't agree with the main elements of his hypothesis.
So why did you correspond with him?
Well, I wanted to examine his hypothesis. But I'm going to, when I write this paper or book on the muscle, I'm going to discuss it seriously. How many tapes have you taken?
It's two. Both 90-minute ones.
I see. Could you do me this little favour? I don't know if it's a little favour. It was a big favour, and you haven't got time to do it. Could you possibly put this into a form which I could take it to the... actually, if you'd like to come with me, we go to the university and try to get it on the internet.
Now you mean?
No, not now. No. Well, it needs typing out first, I think. I mean, can you put it on...
Questions? Well, I could get it onto a disk, yeah.
Yes. Well, these are the questions. You did them before, you see.
Yes, I know. But you expect them not to be answered, don't you?
That's right.
Probably correctly.
Yeah, that's right. But I forecast that, didn't I?
Okay, I'll do it. Provided I'm allowed to put as many insulting remarks as I can to the readers.
No, you were answering.
To spur them on.
Oh, I see. No, you answer it. I mean, if you want to.
No, I don't mean that. I mean, can't I put in insulting comments? Like use spineless gifts or something?
No. Yeah, that's going too far.
Can I make them anonymous and put something like that in?
No, that's exactly the thing one must never do. In science, they're waiting for you to do that. They're waiting for you to insult them, and they can say you're emotional, unbalanced, you know, crazy, you don't know the literature. They can say anything they like. There's a colleague of mine who buggered me at the university. You know what he did? He said that teaching wasn't my strongest point. Now, wasn't that brilliant?
You mean as a sort of review of your performance?
Yes.
It's not very nice, was it?
It was malicious.
Is it true?
Well, I asked everybody who ran any course which I taught, and I had a big pile of 12 letters saying, one of the chap wrote to the Guardian, said that I was one of the best teachers who'd ever taught him. I mean, it doesn't mean anything about one person, but I mean, I haven't, on the whole, I've got quite good, I get 100% attendance in the lectures when I used to go.
Wouldn't insult.
That's right, but I mean, that's what I'm saying. You can say anything you like, can't you? If you get into emotional things, that's why I don't want to tell them they're a lot of spineless gits or crooks or anything like that. I mean, that's irrelevant, really. The questions are irrelevant.
It might be an attention-attracting device as well.
Yeah, no, it won't. It will just attract.
It might attract the wrong sort of attention.
The wrong sort of attention.